So very good afternoon, all of you, and to whoever you all are here. And trust me, this is going to be like, how long is a typical Hindi movie? Typical Hindi movie is 2.5 to 3 hours. That much time we are going to spend today. And trust me, hopefully you will find it as entertaining, let's call it edutaining, as a Hindi film. I'll just bring up my slides. So if any of you are educators, and hopefully all of you will go on to teach your juniors and residents and fellows later on, always remember that whatever you talk should be edutaining. It's just education is pointless, just entertaining is pointless, right? Try and be edutaining as much as you can, and hopefully, you'll see a little bit of a glimpse of how edutaining lectures could be. So I generally like to start my presentations with an inspiration. So today I thought, because it's a research-based topic, and I do talk on a lot of different aspects like AI in medicine, and then health and nutrition and retinal diseases. But today we are talking about research. So I thought this is an inspiration to begin with. The questions you ask define who you are and not the answers you know, right? So at an early stage of your career, you may find it a little bit, not very deep, but as you graduate, you will realize that this is, more and more important. So now I'm going to show you a couple of photographs of some people who asked very deep questions. And, we'll delve a little bit into the questions of, the kind of questions they asked and why they perhaps won Nobel Prizes for that. So the story is about two friends, so one is Barry and the other is Robin. The one on the right is Robin. How old do you think Robin looks? They are Australians, by the way. This is a photograph from the 1980s. So how old do you think Robin looks like, the one on the right? Anybody? 55, all right. Maybe 50s to lagi raha hai. He's not looking young. So he is a medicine resident, internal medicine resident in his early 40s. And this is a very young pathologist in his late 20s. Right, so they started asking a question: why do people get burning in their stomach after they eat? What is this gastritis? So all of those questions were asked, and they came up with a hypothesis that there may be an organism which is surviving that gastric acid pH, and that is what is causing this disease. And they were shot down by a lot of people. People shot down saying, oh, the pH of 2 or 1.5 cannot be tolerated by a pH of 1.5 or 2 cannot be tolerated by any any organism, right? So so they persisted with this theory. They presented it in many different conferences, and so that work started in the early 1980s. By 1992, that's about 12 years after they started that work, it was recognized by the NIH that that Helicobacter pylori actually causes gastritis. And why I am telling you this is because they had to actually drink a glass full of H. pylori, cause it in their own stomachs, do an endoscopy, and then prove that H. pylori causes gastritis after about 3 or 4 weeks of inoculation in the stomach. How many of you would go to those extents to prove your hypothesis, right? So the question we began with is, how far would you go to ask a question, how deep that question should be, and how much you should pursue with it. And because of this, they were awarded with the Nobel Prize in 2004, and that's about 24 years after their seminal work, right? So from Nobel, when you look at Nobel Prizes, the mean interval from your inception to the Nobel Prize is more than 40 years. So they got a Nobel Prize within 24 years. So that's pretty seminal work, and we know that this is really important, and H. pylori does cause H. So the whole thing started out with is how do you ask questions? So the inspiration is that if you ask the right questions, you will get the right answers. And Professor Amod Gupta, I'm going to show you a quick glimpse of what Professor Amod Gupta has to say about asking questions. So I need Is there any special message for young researchers who are watching this episode?
For the young researchers and young doctors, my parting advice would be to not stop asking questions. What I say is, before you learn the answers, learn to ask questions. In my paradigm, learning questions, how to ask a question, how to frame a question, how precise you should be in asking that questions and asking your query. I think that's what, with the availability of Google and such search engines, it's so easy to find whatever is known about a particular subject, about a particular aspect of a question, and you need to find out the outer boundaries of knowledge. You need to figure out what questions have no answers, and those are the questions you need to chase in your research. They are the questions which you should frame your research grants and research questions, and try to figure out the answers. And I'm sure all those answers that you find, all those papers that will emerge would have a huge impact all over the world.
So I hope all of you have heard about Prof. Amod Gupta, right? How many of you know him, and how many of you don't know him? All right, I mean, I'm sure. So he was, he's been the pioneer of asking questions about ocular tuberculosis, and we know so much about ocular tuberculosis because of the questions that he's been asking for the last 20 years, right? And Prof. Rupesh is with us, and he has now asked a lot of other questions about ocular tuberculosis and come up with a COT study, and now we know more about asking questions. So for lack of time, I'll just go towards not introducing too many things. So I did my research training at Johns Hopkins Hospital, and really what I learned there was how to frame a question and how to refine that question. Some of my images from there, and I have a very esteemed group of colleagues to talk to you about different things, including study design, which Dr. Apurva here with us will talk. She's from Joshi Eye Hospital. She's a vitreoretinal surgeon, a very accomplished one, and she has done a lot of good research in vitreoretinal diseases. Professor Dr. Shayan Basu unfortunately won't be able to join us today, but he was going to talk about paper writing, which I'll cover. Dr. Akshay Nair, he's also going to join us in a little while. He'll talk to us about case reports. He's an oculoplasty surgeon in Mumbai. And Dr. Shamrat Chatterjee, he's a practicing cornea refractive surgeon at Joshi Eye Institute, Raipur. So this is the Ghibli version of this. I thought, let me see what ChatGPT can throw up, and it is throwing up a lot of versions, but this is the one I thought will be appropriate to show you, right? So before we dive very deep, let me try to bring out how research is very, very important as we go ahead, not just operating on good cataracts and giving intravitreal injections, but this is how medical practice might look at like by 2030, where a lot of automations are going to come in, right? So the keyword here is automations. A lot of the things that we do today are going to be done by machines. So we are really in the phase of sort of AI assistance at present, right? But then partial automations and perhaps full automations may come in. And ChatGPT is learning all the time. There are many devices. This is just one example where a device can actually throw up a lot of parameters from your eye, including your systemic health in about 90 seconds. So Google has somebody who is called the Chief Medical Officer, CMO. Google, we know it's a technology company. So what is a medical officer doing at Google? This is what he's doing. He's managing this very large building which is called the Google Medical Center, and it's not a hospital, but they're working on health tech, right? So this is very much coming in, and what are we going to do then, right? So you will have to delve into validating some of these new products that come in. Unless you are aware of research and methodologies and analytics, you will not be able to sort of participate in all this. And so a lot of data I had put in to show you that machines are doing as well and sometimes even better than what humans could, and I keep posting about these things. So this is an AI model which actually is telling you which injection will be better for the patient based on a lot of other parameters, and it is under review, and I just reviewed this paper, so therefore there is not much information about this, right? So there are 4 treatment groups which have been proposed to the AI, and it has been given all the information about the patient, and the AI is actually telling if you are on this, please switch to that and other things, right? So that's how it's going to actually change. So we might actually look like this. So it's everywhere. It's good for patients definitely, but it's not so good for the physicians, as you can realize, right? So some good books which have been written on this is 21 Lessons from the 21st Century, which talks about med tech or ed med, right? So medical education and technology in terms of just education is going to change a lot. So we can be prepared in terms, it won't happen overnight, and we can assist in this evolution and be part of it, right? So to be part of it, you really need to, buck up, right? So just learning clinical skills is not enough. You need to, find new ways to equip yourselves to be ready when, these things are going to come into vogue. So, this is another great book by Michael O'Grady. It's a very short read. So it talks about, think smart, work smart. So he says, first line he says is don't work hard, work smart. And that really means, working even harder. But then that you realize at the end of the book, right? So how you can be prepared is you can train yourselves with some amount of analytics, some statistics, which I will actually demonstrate. And you will see the power of AI doing all the statistics from the protocol T of the DRCR network, no less, right? We'll uncover some trends which no one has seen before, right? So, simple question like, does every patient with diabetic macular edema need a loading dose? And most clinical trials will say yes, but I'll show you some from their own data set which will say 28% perhaps don't need a loading dose, right? So you can cover many things like that. These data sets are all available. So you need to be good at medical research and be prepared for the tomorrow. So this is the overall roadmap that we are going to cover in the next 3 hours. So you need to plan and execute a study. And to do these things, you need to do your own studies as well, right? So then you need to analyze and write it up, and then review your own work, submit, and then eventually get published in a journal. And the whole endeavor of the next 2.5 hours is going to be how we do this. So I started my journey way back in 2008, and that's how I still look like way back in 2008. So you can actually, I've done a lot of courses at Arvind, at CMC Vellore, and then at Johns Hopkins. And I realized that you need to understand a little bit about statistics, about writing, and then I learned a little bit about editorship when I was at IGO, but that is not always important. Writing and doing analysis is important. So you can get trained. There is a lot of material which is there which can help you get trained. So because of lack of time, we'll just go ahead. I'll quickly show you a couple of slides on why you should do research in addition to just being relevant. The mnemonic I've made for it is called FAMOUS, where F stands for fame and recognition. As you can understand that, if you've published something on niche work on a particular disease, you will have fame and recognition. A stands for patient care. So a lot of your research could be pivotal, right, in terms of how it's been looked at, and how people are adopting. So some of your papers may be read by 1,000 doctors, and they may apply it to 100 patients each. So you will actually reach out to like 1 million people over a lifetime. So then it could be that that penetrative. You will make yourselves a good clinician. Who doesn't want to be a good clinician, isn't it? So there is a lot of back and forth with the editor and reviewers, and that will help you troubleshoot. So whenever you're actually sitting real time in front of a patient and you're not able to figure out, your mind is already agile. So you will become a better clinician. A lot of papers could influence policy and decisions, right? So, not only physicians read your papers, but IAS officers read papers, judges read your papers, the media sometimes reads your papers. So a lot of decisions, including the NPCB, the amount of money which is allocated to the NPCB comes from some of the papers that we write, right? So policy decisions are impacted. Funding and grants are part of this whole process, where if you write enough good papers, you are eligible for a lot of funding and grants. And then the personal index, that really means citations. How citations, basically your job as an author is done because the paper is now published, but the paper's journey is just beginning, and it's an eternal journey, isn't it? So the paper is going to live on and on even after you are not. So people will read those papers, and if they find it helpful, they will cite it in their own, papers. So the more citations your papers have, the better penetration you are having. And the Stanford University does this whole study, if you know about it, right? So they bring in a list of top 2% global researchers, and that is entirely based on citations, right? So that's basically the personal index. This is a lecture I have on YouTube if you want to scan it with your cameras. Here I talk about some commercial aspects of research. So basically, how can you generate revenue? And sometimes you could generate so much revenue that you may not have to see even a single patient, right? And so you could scan this, and you will have a YouTube link, and you could go ahead with that. So so this is just the primer of why you should be doing it, right? So that why is more important. So I'll just end with a quote from Simon Sinek, who is a great orator. If some of you have heard about Simon Sinek, so he talks about two ways of marketing. One way is where say a manufacturer like Windows tells you that, buy my product because it has this RAM, this GB, this resolution, and so many other things, and therefore you should have it, vis-a-vis an Apple, which then tells you, it never mentions about the RAM and the speed. It just says, आपको moon पे जाना है, मेरा device लेके जाएगा. If you want to do this, my device will enable you, right? So that's the why behind how they market is so much important. So unless we understand why we want to do this, it doesn't make sense. So after we finish why, let's go ahead and learn more. So Dr. Purva will talk to us about study designs and how do you actually bring it into your into your own practice. Dr. Purva. Yeah, so in 2017 there was so the question was, has any paper, do you know, has impacted policy and decision, right, so far? So there was a paper from Dr. Praveen Vashisht in 2017, which actually reclassified, blindness in India, which was based on the WHO definition. So WHO talks about 3 by 60 as the cutoff point, whereas India used to talk about 6 by 60. So if you use the WHO definition, then the number of blind actually go down significantly, because you're looking at a 3 by 60 in the better seeing eye as the definition of blindness. So once that comes, the media also reported, oh, the number of blind in India has gone down. But actually just the definitions had changed. And now based on that, the NPCB has taken note of that, and then the funding for the NPCB has actually gone down. So that is one example that I know, and there are several others internationally. Say one example is, there were studies which looked at phacoemulsification reduction causing reduction in intraocular pressure. So there was this ultrasound causes micro changes in the trab meshwork, and how this ultrasound is making the trab meshwork more porous. But then subsequent studies talked about SICS also causing drop in IOP. So then the ultrasound perhaps has no role into that. So the people who are working on the trabectome and some of these instrumentations actually lost a lot of funding from the NIH. So then there are a lot of examples of some of your studies which could influence policy, decision, funding, and so many things.
So just to give very quickly 2 examples, if you allow me. One is the trauma registry which we set up. We worked with National Human Rights Commission in 2022 to set up a white paper by NHRC involving all the ministries. And now there is a PIL filed by Dr. Natarajan based on the trauma registry data of fireworks. So we are filing up a PIL against the government of India of stopping and all. So this is how it can be helped. So cornea people working on chemical injuries, Chuna and all those things. And Dr. Geeta here has done a lot of work on that, and then I'm sure there are a lot of people work. So this is how you can probably impact. The second thing which we managed to impact in India is the ocular TB study, which Dr. Sengupta sir mentioned. Is through that we are able to get into the ICMR as a form of extrapulmonary TB, ocular TB being recognized. So first time ever in many years, now ocular TB is listed as, and there is a guidelines about how to manage ocular TB in ICMR. Otherwise, before that, it was never listed as that. So these are just quick 2 examples.
Yeah, I think.
So I think, I didn't know about that litigation, that PIL thing, but probably if that study gets published in IGO, probably that would also tell you about the opinions of probably around 200 odd ophthalmologists all across the country.
So this is where Dr. Sengupta's thing is so important, that when you say why to do research, because sometimes you do not know your contribution can make such a bigger impact. Because we do the things when things affect our family and our home. But when you do this thing, it will affect the entire community and the nation and the global at large. So therefore, it is such an important thing which Dr. Sengupta has mentioned, is about why to do research. And your paper will be bigger thing into that larger pool of things.
Just to add on to all this, right, if your question was, which was a publication which contributed to a policy making decision. That's different from evidence in literature talking about a particular category of morbidity or events arising from the morbidity. There might be many, but not all policy decisions are going to be based on that. So I think as of now, I know the policy making decision is Dr. Praveen Vashisht data, which has, altered the Indian government's look at the category of blindness, and we have a new categorization on that. The others are data which are existing on the particular morbidity as well. So they are not really, getting into an ICMR is not a policy making body. That's something we will have to understand. ICMR is a council which conducts medical research. Yes, a research which is conducted out of an ICMR protocol or a team has a higher validity given the category of submission the article is. For example, a letter to editor submission is not going to carry much of a weightage. A commentary is not going to carry much of a weightage vis-a-vis an original article. If you look at an original article, again, an original article, which is a retrospective study, will have several flaws in its methodology, whereas if you've done a prospective RCT, it carries a different. Again, if you're looking at review articles, if you're looking at a systematic review vis-a-vis a meta-analysis which is done systematically, again, the weightage of these articles and I think the standard and the quality of recommendations they give is also different. Moving on to what Dr. Samrat was saying, see, 200 ophthalmologists out of a country which has 26,000 ophthalmologists, it's a minuscule opinion. You might have many others whose opinions could be different, who were perhaps not accessible on that WhatsApp. So it is not reflectable of the entire community. So the way you interpret research which is published is very important when you arrive at a particular decision or at a particular way you are going to conclude on a particular morbidity. That's how you would learn to interpret research. Not that all that gets into publication in any journal needs to be reviewed. That's what we as reviewers have to teach ourselves. How would you probably critically review a publication which has gone into print? And that's what we all learn, and that's why journal clubs are very important, because it teaches our residents how you will have to critically analyze the statistical data which is being provided to you.
So I'll just like to provide a clarification about, not exactly, I know my words might not be correct about policy, but what are the steps?
I'm sorry, not to contest what you've said.
What are these
this
steps?
is just to give the right context
second
in which
example
we
of
have to interpret.
phagoemulsification is not a policy level decision. So first example I can understand, but the
You
second
probably you said,
I'm just trying to say that because a group of researchers, if they do research, how bigger impact it can make beyond a thing. So sometimes people think about a single case report, but there are less report, and then how does it make an impact about clinical decision making? There can be multiple steps. Clinical decision making, does it influence? Second level is thinking about a policy, because first step, because you're not going to go in policy making overnight. It has to be a step building thing.
I totally
you start
agree,
thinking, and
I
then it will build towards that.
Relating to trauma, we have several publications which are now coming in IGO. It's not only chemical injuries, laser light injuries.
Yeah.
Now I think laser light is one of the important things in what all event management teams, and it's starting to appear in a higher quotient in almost all events here. And laser light induced retinal injuries, macular injuries is a reality, and we have more and more publications which are coming into IGOCR in this. So that's probably another health issue which we have. And a second one which I did present in one of our later meetings here was that people use their hands for fireworks and firecrackers, and that not only causes damage to the eyes, it also causes damage to the hands when they hold. And I recently landed up operating one such patient. I had to do a bilateral therapeutic keratoplasty on that child who was holding the cracker in the hand, and he lost both his hands, and he can't even use Braille for his rehabilitation. So that is, I spent that 1.5 hours, 2 hours, I had to do both the eyes, and it was a very sorrowful, and I had the lowest of the moods of those. So that's particularly things which probably when we publish in our journal, we can highlight to the community, and yes, and these would probably make an impact on policy making decisions. That way what Dr. Rupesh says is absolutely right.
Thank you so much, ma'am, and Rupesh sir. So I'll invite Dr. Apurva to talk to us a little bit about study designs.
Thank you, Sabyasachi sir, for having me in this meeting with such illustrious people. And my talk is going to be about types of clinical studies and how to plan a study. So these are the types of studies. I'm going to talk about each one of them. Let's start with case control study. Here it's very important to know that the starting point of this study is the disease. So the study goes in a retrospective fashion where the deceased patient and those with no disease are going to recall about their exposure. So cases are the group of people with disease, and control group is the one which does not have the disease. And this type of study basically determines the odds ratio. That is the odds of getting the disease after an exposure by the odds of the disease without the exposure. So basically case control study answers the question about what happened. Now let's look at this example of a retrospective study wherein groups with different known outcomes are compared based on a causal factor. So let's look at this study, which is a case control study of risk factors for AMD. This was done in France, and you can see that this determines the risk factors, enrolled patients with bilateral AMD. So the starting point of this study is having the disease already. And they were matched with controls who didn't have the disease for sex and age. Environmental factors and histories were screened. So now we have 1800 controls and who are compared with a similar number of patients with AMD, and they found 6 major risk factors of hypertension, coronary disease, hyperopia, light colored iris, lens opacities or previous cataract surgery, and coronary disease. Had atrophic disease. Now let's see the drawbacks. Here the patient may have recall bias. AMD patients are more likely to remember longer sun exposures than the control patients, and there will be a selection bias also because patients with the disease may choose to enroll in such studies. Now coming to cohort study. Cohort study starts with the exposure. So it's a prospective study. Now if you look at smokers versus non-smokers, like it was seen in the Blue Mountain Eye study, here the exposure patients are the ones that are going to be studied prospectively. And these both these groups, this is the case, this is the control. And now you can study multiple outcomes. You can study whether they have more nuclear cataract, whether they have younger age of cataract, whether there is more late AMD or higher IOPs. So the advantage of this study is that you can study multiple outcomes because the disease has not happened yet. So this answers the question of what is going to happen, and it tells you the probability. The probability of disease in the exposed group divided by the probability of the disease in non-exposed group. So this is an example of a cohort study where they studied the incidence of myopia in Singaporean children, and they found out that for every unit increase in myopia, parental myopia and IQ scores had a higher probability. So the drawbacks are obviously there because follow-up runs in decades. It is time consuming, it is expensive, it has a risk of attrition of subjects, and subjects may change their preferences. So smokers can choose to quit smoking, so they're no longer in the cases group. Can a cohort study be retrospective? It can be when we do a retrospective chart review. Here if you see this demographic which wears contact lenses and were first fitted at 8 to 12 years, they had 1,000 children. And if you look at the adverse effects of what they had, you can actually see that this is a retrospective chart review which started with the exposure and went on to study the adverse effects over several years. This is a cross-sectional study. It analyzes data at a single point in time. It tells you the cross-sectional data. It tells you prevalence at that point of time, what is happening. This is the next type of study, which is randomized control trial. Here you can study the causation, treatment, the diagnostic tests that are there. So patients, the subjects here are randomly assigned to either test group or control group. Like this, for example, you can test for what happens when you inject a patient with a drug versus a patient who has not been injected with the drug. Important that the groups should be similar in gender, in the disease, in education levels, for example, geography, ethnicity. And blinded studies will reduce bias. Here the participants can be blinded, researchers can be blinded, and best is even if the data analysts are blinded. So randomized control trial can actually evaluate the efficacy and safety of a new potential treatment, and you can tell the differences whether they are statistically significant. So to put it in a nutshell, so did the investigator or the researcher assign exposures? If no, then it becomes an observational study. If there is there a comparison group? If there is no comparison group, then it's a descriptive study. If there is a comparison group, then it is an analytical study. Now, if you look at the direction, is it has the outcome already occurred, and is it a retrospective study? Then it becomes a case control study. And if it starts with the exposure and studies the outcome after several years, it becomes a cohort study. And if the exposure and outcome are being studied at the same time, then it's a cross-sectional study. Now, if you look at experimental study, is there a randomized allocation? If it is not randomized, then it's non-randomized, and if it is, then it becomes a randomized control trial. So a crossover study is when they are divided into two groups, and then there is a washout period, and the patients or the subjects are crossed over to the other group. So this is a variation of an RCT itself. Again, it determines causation. Each group receives either A or B for time X, and then there is a washout period where they switch, after which they switch the treatments and continue again for time X. Case reports, all of us have a lot of case reports and series. A number of more than 3 subjects is considered a series. It documents a single patient response to disease or treatment, or a small group of people. So coming to planning a study, how to formulate a research question, how to search for literature will be covered in the next talk, and how to access that literature. How do I conceptualize? You read and read. You do more detailed literature searches. You attend seminars, conferences, webinars, discuss the subject area, listen to more people and ask questions. Finding the focus of research is very important. Which aspects of the discipline interest us most? What have we observed that you have questions about? And what articles have you read that have raised questions in your mind? So a good example of finding out what is being published or what is being published is to subscribe to e-table of contents. Just write your email address here, and you can sign up for a newsletter which tells you the table of content for every month of the journal issue, every journal issue basically. So it has to be original. It can be a novel technique, a novel clinical finding, novel use of drug, finding a novel mutation. This is an example. So I'll just come to that example again, which is why I skipped the slide. Background questions are very broad. It answers broad questions like what, which, when, why, where. The foreground question is the one which gets down to the specifics. What are the treatments of PCV is an example of a background question, whereas the foreground question gets into specifics. In a patient of PCV with pachychoroid, will adding PDT to intravitreal aflibercept reduce the number of rebleeds? Look at the specifics in this question. So framing a research question is based on the PICO, which stands for patient or population to be studied, I for intervention, C for comparison, and O for outcome. PICOT adds 2 T's, wherein which type of question you are asking is one, and the type of study is also one. So you have to find out where are the gaps in the literature, like you saw in the video of Professor Amod Gupta. Here, what is not being looked at, what is not being done, who is not being studied, and who is not being compared. So let's look at these research questions. Patient or population can be chronic CSC. The intervention, like let's go PICO. Intervention can be subthreshold micropulse laser. Comparison can be with photodynamic therapy, and outcomes in terms of final visual acuity. This is an example of a therapy question. So the next PICO, let's see. Patient with chronic CSC with obstructive with sleep apnea, which is an exposure or an etiology. Comparison with no sleep apnea, and the number of recurrences is the outcome you want to study. This becomes an example of an etiology question. So we have to keep it simple whenever we plan a study, right? Specific aims and objectives. Why do this project now? What are the immediate benefits of doing this study? Are the outcomes measurable? Is it practical? Is it logical? Is it evaluable? This is the hierarchy of the study designs which I spoke about. This is borrowed from Sen Gupta's Research Academy. So the bottommost is the animal and laboratory studies. Next is the case report, and then case control studies, prospective studies, randomized control trials, and the highest level of evidence is provided by meta-analysis. So you have to know the ideal study design. For example, a therapy question, you should have an RCT. For etiology question, the best is case control, and it should be feasible. Like if you are in a smaller center, it's not any use of doing something like a retinal sensitivity or something, you know, high flying. So failing to plan is planning to fail. I thank you again for giving us this opportunity.
Thank you, Dr. Apurva. And so that was a good overview on, what is prospective, what is retrospective, and all of these. So, when we talk about randomized control trials, though, it's perhaps the most difficult study design to pull off. And, you will see more and more study designs in terms of, I'll call Dr. Shamrat to just set up your slides. So, so you will see more and more about non-inferiority study designs. That's what most of the clinical trials nowadays do. And so the traditional approach, say, when streptomycin was being tested for tuberculosis, which was actually the first ever RCT done, so it was treatment versus placebo. That is, nothing is available, right? So currently we don't have that, right? I mean, there is always, there is a treatment for every drug or, every disease which is there. So you can't actually treat a neovascular AMD patient with nothing, right? So that will be totally unethical. So the question that we want to ask now is this drug superior to what is existing? But that is, that boosts the sample size very much, and there are a lot of problems with doing of these. So what we now want to do is ask the question, is this new medicine as good as what we were using before in terms of the primary outcome? And maybe it is slightly better in terms of a secondary outcome. And so the non-inferiority study design really starts with the basic question, are the two drugs the same? And the superiority study starts with a completely different question, is one drug better than the other? So when you think about the opposite question, which is called the null hypothesis, and which is all dependent on, based on which we interpret studies, do sample size calculations, and many other things, you'll realize that the null hypothesis is exactly the opposite of your hypothesis. So in the superiority studies, you will say, is drug A superior than drug B? And the null hypothesis is, no, they're not. They're the same. In the non-inferiority, you're starting with both are the same. And therefore, your hypothesis is one the null hypothesis becomes is one better than the other, right? So they are very different in terms of the study design, in some of the statistics done, in terms of the interpretation and all of those, right? So we won't have a lot of time to get into the nitty-gritty. So an additional thing that you might want to remember is non-inferiority study designs in randomized trials. So I'll now invite Dr. Shamrat Chatterjee to talk to us a little bit about literature review.
At the outset, I would like to thank Dr. Sengupta for the opportunity. And thank you all for finding time to attend this. And so literature review, that's very important, because oftentimes when we suddenly, we get a eureka idea, then we start jumping into the study. And when we get the results or when we conduct the study, then we go into the literature to see, because now we have to write a paper or do a presentation, so we need now to compare. And when we start comparing, then either we find that we have done all the work, has already been done, or whatever has been done in an incomplete way. So it is always, because this session is only on research, so the literature review that I'm going to talk about will be focused mostly on the research part. But the literature review is also important if you have to do a clinical decision or a clinical treatment, so in the clinics. So it's not only that a literature review is restricted only to research, but it comes in different areas. And when you apply it to research, then you need, the moment you have an eureka idea, ideally you should go back to the literature and see whether somebody already has that eureka idea or not. And then you need to read those articles, and if you suddenly say that, okay, it doesn't have much of uniqueness, then probably even if you send it to a review, the editor, as Professor Vanati has said, would not find it very interesting, and it would get into a rejection. So it is always better to do a literature search at the beginning. And also, you need to do it to design your methodology, because somebody would have done part of it, and if you do a literature research search, then the methodology part becomes much more clearer. And later on, when you are writing the manuscript, you need to compare your results, find out the flaws, find out the gaps. So all those things you really need to do a literature research. So that is a primary step before you jump into doing a study. So there are sources of literature. So traditionally, literature sources have been defined as primary. That comes from the journals. It comes from a secondary source, like a meta-analysis or systematic review. And later on, once these are inculcated into clinical practice and a lot of people have thought about it, those findings have been published and finalized and polished and refined, it comes into textbooks. So if you're going to do a literature search, it is always better that you go into the primary source, the journal, rather than the textbooks, because there is a gap. So by the time it comes into the textbooks, it's quite possible that some concepts or some treatment may have changed. And already Dr. Apurva has shown this pyramid of sources. And of course, the cohort or case control studies lie at the bottom, but it doesn't belittle those reports, because unique findings would not come in a large case series. There would be a single case report. So a single case report of, say, like a bilateral cracker injury, where there is bilateral forehand and hand injury, that other people may have much more cases. A collaborative effort may bring out a case series. So even though the case report or these studies are at the bottom of the pyramid of literature search, it doesn't mean that they are to be belittled. Of course, a systematic review or critically appraised review and all those, or RCTs, are higher in the pyramid. And we all know where we get the articles from. Either we get it from journals. The journal articles, abstracts, they are collected into some depositories like the PubMed, Scopus, Ovid, or Cochrane, then the books, and also sometimes you can get data from the IHO or ICMR, NPCV, and even data from the NCB. So suppose you want to do something on the population, you want to find out the prevalence, you need to go to the census data to find out your age-adjusted prevalence and all those things. So all those things also come under the purview of a literature search and is important in research. So how do we do a literature search? So there are lots of web-based tools, though we are much more familiar with few, and this is an exhaustive list, so I don't have time to go through all those things, but there is a lot of search engines that are there besides PubMed, but we are all familiar with PubMed, because PubMed was one of the first of these kind of search engines that allowed us to delve into literature, and it has been there for more than 6-7 decades. So I'll do these examples through the PubMed. So PubMed was actually initiation from the National Institute of Health, and it started off as Medlars Online in 1964, and all of us in fact are familiar with it. And it has different components. It has a Medline, which gives us the article database. It has a PubMed Central, where you can get the full literature, full text, and you also have a bookshelf where you have the archives for chapters and reports and other databases. So a typical PubMed interface comes like this, and most of us restrict our literature search into the method 1, because this is what pops up the moment you put PubMed in Google or on the search engine. But there is a method 2, which is a refinement of the first method, an advanced search, and ultimately you have something which is known as the mesh database, which is much more refined, and I'll take you all through this entire 3 processes. So it is always a good practice to do a login into the Medline, into the PubMed, because the login, most of the time we don't do a login. So what happens if you do a login is this search is saved. And once this search is saved, you can then again go back later on, maybe after 3-4 months, and use that search keywords to find out if within the period of your research something else has cropped up or not. And these are few basic things like the Boolean terms. Most of us know what the Boolean terms. So you do it. You use add, and, or not between 2 keywords. So add would include both the keywords, or would have abstracts or titles or the full text where both the keywords feature either one or the other, and not which we use it less where we want to leave out a particular keyword. So that also I would cover it. So this is a kind of a algorithm that I took from this article. So if you have a type of question, and if it is, let's not talk about the therapy part first. Let's talk about the research part, because if you want to look at the diagnosis or at the prognosis or at the etiology, then we need to do a PubMed clinical search. This is the general query that I will take you through. And then if the data is not there, or then you can go into the other databases. There are other databases, but the other databases like AI, which I'm going to cover briefly, and I think Dr. Sengupta is going to cover it in larger details. Sometimes they have their limitations. So the other databases would come with a rider that you need to really evaluate those databases. Otherwise, if you're going for therapy, one of the most good research databases that you have are the Cochrane library. Sometimes the Cochrane may not have that, and then you can go into the PubMed clinical query or the systematic review. So there are other tools there within that. And if there is a no systematic review, then you can either go in for a RCT, search the general or the central registrar. Even there are, we all know that nowadays if you have to do a randomized clinical trial, then you have to register that with different bodies. Like in India, it is it's with the drug controller, CTRI. So there are different repositories. So it may be that the study that you want to conducting and RCT is going on, and that would either give you the design of the RCT, or it would also sometimes give you the preliminary results or the final raw results before it goes into publication. So let's start off with an example. And so, as we all do, we ask a research question. So the research question that I've chosen is, what is the role of topical corticosteroids in the treatment of bacterial corneal ulcers? So we all know that bacterial corneal ulcers, the topical steroids are no-no. So we want to find out whether if we give topical steroids in bacterial corneal ulcers, is it good? So this is the research question. Like Dr. Apurva said, you need to ask the research question at the onset. But we need to break down the research question into appropriate search words. So from the research question, we have broken down this. Role is a verb, so adverb. So it doesn't actually is a keyword. So the other keywords are topical, corticosteroids, because topical, why? We are thinking about probably a type of drug administration route. Corticosteroids, bacteria or bacterial, and corneal ulcers. So there can be others like microbial keratitis or keratitis. And then we put it into this is the method one, the general box, that is we all are familiar. The moment you do PubMed, this thing will come up. And then you include all these keywords. Now earlier, even if you don't include the Boolean terms, now the PubMed would include those Boolean terms for you. So including all those terms out here, topical corticosteroids and bacteria and corneal ulcers, what kind of a result we get? So we got around 106 results. Because this was a slightly niche kind of a research question, the results are 106, and probably it gives you enough time to go through the results. But sometimes you can come up with thousands, more than 10,000. So do you really have the time to scroll through all the 10,000 articles to find out articles that are of interest to you? And the previous versions of PubMed actually did not have many of these filters. Now the recent, they have filters, so you can filter according to the time period. If you want recent articles, if you want all the articles, then you have the type, what kind of an article you want, and there are other filters out here if you want to do a clinical trial. So even after clicking on the clinical trial, so we get around 10 results. So this is after we have, we have removed all the articles that are either reviews, case reports, or other kind of case series, and just included or opinions, editorials, comments, letters, and included only RCT. So by doing this, you narrow down the 106 to at around 10 results, which are much more focused and much more specific. What does the method 2 or the advanced search does, it does more or less the same thing. But it now gives you lots of options. So this option of all fields, so there's an option of all fields out here. So this all fields would give you whether you want these keywords to feature in the author name, title, abstract, main text, so or other databases. So you can filter off all those things. So we want only, say, I usually just select the title and the abstract. So most of us, we know when we write the title and the abstract, we would include the keywords there so that the keywords become more searchable. And then we use, again, we put in the keywords that we had selected, topical and corticosteroids and corneal ulcers and bacteria. And even when we do that, we get around 102 results, just 4 short. And again, when we do the filter, we land at about 10. And suppose we want to do the same exercise with mesh, like even say 10, we don't want to go through all the 10 articles. We want to just look at if there has been any good trials that have seen whether corticosteroids benefit patients with ulcers. So we go to the mesh term. And when you go to the mesh term, so there is a different search engine for the mesh. The first thing when you search with the mesh is you want to find out the keywords that you have selected for your study, are they mesh words? So let's see. So first is topical. So when you put it into the search box out here, mesh and topical, you find that yes, topical can be a keyword for that. You have administration topical drug. So topical is a mesh term which can be included. So we put it into the PubMed search builder. So we click on that and it comes administration topical. So next we go at corticosteroids. Now the moment we put corticosteroids as a mesh term, we see that corticosteroid is a mesh term, but it comes as an adrenal cortex hormone. So now we need to modify the, please come in. So the corticosteroid is not a mesh term, but steroids is a mesh term. So sometimes we need to modify our search terms also to find if they are mesh. And so here you see steroid becomes a mesh term, so we add steroids to the search builder. So we have done topical, we have done, so corticosteroids not, so we have done steroids. Now let us see if corneal ulcer is a mesh term. Microbial keratitis is not a mesh term, but corneal ulcers is a mesh term. So corneal ulcers comes and then you can add it to the, again, the search builder. And lastly, bacteria. So bacteria is a mesh term, and when you add it, so what do we get? We get 10 results. Now, if you look at the 10 results, from those 10 results that we had got earlier, now we have got into 9 results. And when you see the 9 results and you see the type of articles that you get, these are the niche articles, the most quoted articles, most cited articles on the use of topical corticosteroids in bacterial corneal ulcers. So you have these SCUT study. So you have the SCUT study, which has actually, for the people dealing with corneal infections. Based on that, now we are much more liberal with the use of steroids than we were probably 10 years before. And here also you can again use these modifiers like clinical trial, and then you can get all these kind of filters. And so that is how you can actually use the mesh, the Medline, the PubMed has 10,000s and thousands and thousands of articles, and using the terms and things like that, you can filter it down to the ones that are very, very relevant to you. But is PubMed the only thing? Since last 5 to 7 years, we have other search modes like AI. And even in this conference, so there was a session on education. There were 4 or 5 speakers who were talking on AI. There are a lot of AI into this thing, and I think Dr. Sengupta has also is going to emphasize on the use of AI. So AI is also used as search modes. And how AI has come into search mode, it has come through Google. So like the simple thing is that if you can't find it in PubMed, you Google it, something or the other is going to come in. Now Google is also, the earlier versions of Google and now the versions of Google are different, and Google also use some kind of AI and much more relevant AI to us as medical professionals now. And when we put the same thing as a sentence, because it takes the natural language mode, and you can see it also gives us studies, and even the SCUT study is featured there. The AI, there's an AI tool now, and this AI tool has, it gives you a summary, and it also gives you links, and through those links you can get through the articles. See, in that Google search also you have the steroids in corneal ulcer study that is featured. Probably this would not have featured in the top maybe couple of years back when this tool was not available. So Google has been actually improving on their search engines for a very, very long time, and it has benefited us also, and we are seeing the benefits of it right now. There are other AI search engines. I've just taken one because of shortage of time, something known as Perplexity. And there also you can see that this AI also, when you give in the same kind of a research question, it will give you a summary. This is very helpful in your in the clinics. Suppose you encounter something and you're not very familiar with it, you can use these kind of tools. It gives you these key benefits in severe cases. So one of the SCUT, and this quotes this SCUT study. So the SCUT, one benefit was that in more severe ulcers, if you give steroids, the outcome is better. The natural tendency for us as clinicians is that if it's more severe, probably you would not treat it with steroids. But in Pseudomonas and large ulcers, it found. It also gives us more findings because it has taken now all these SCUT studies, and it gives us specific things like in nocardia. Steroids given to nocardia ulcers in the SCUT study, there was worsening. So it warns us about that. So this is a very relevant tool for literature search. Like there are other AI search engines also, but it gives us summaries, clinical guidelines, recommendations, the last clinical message, and this can actually, within few seconds, you now have a treatment plan for your patient. You have a large ulcer. They have said it has grown Pseudomonas. Now you won't be hesitant in treating the patient with ulcer. Now it's a crenated margin, and the microbiology report comes as thin filamentous bacteria, could be nocardia. You take a step back saying that no, I would not like to treat this patient with topical steroids. But is AI the end? No. There are advantages that I've already talked about, that it handles natural language questions. You don't have to search for keywords or see all those keywords. It summarizes important points. The current tools gives you the important points like it gave us in the Perplexity example that I showed. Very quick, within few seconds, you are going to get what you have asked for, but it has limitations. I think you're going to cover more of the limitations also. So the basic of AI is how, again, like we say in computer, data is what is the data that you put into is what you are going to get out of it. So AI also does the search, and if the open access articles are not there, then the SCUT studies are in open access, so it has been able to retrieve those. But if there are key articles that are not open access, then the search engines would not give those things. So then you'd land up, maybe sometimes you can get information from predatory journals or other journals which are non-peer reviewed, or paid articles. So you need to be slightly careful on those things. So over time, I just wanted to show this. This is how the literature search has actually evolved over time, and from books to this kind of publications, and ultimately now we have the AI and Chat GPT. So whenever you do this thing called literature search, you need to be patient, you need to be diligent, you need to actually assess the source, whether it's trustworthy, peer-reviewed, from a good journal. You need to look at the accuracy if some things are off, and sometimes you also need to look at the bibliography at the end of chapters to get some key articles, and then finally do the search. Thank you.
Thank you, Dr. Samrat. And till I set up my computer, are there any questions for Dr. Apurva or Dr. Samrat before anybody?
Studies that we do, is there any time frame, like this many days, months or years. Say sometime we take a very rare disease and I want to tell that the outcome of this disease between this period as a cross-sectional study. So can I take it as like for so many years? Is it possible or any limit is there?
Basically, when we are studying the exposure and the outcome at the same time, then it becomes a cross-sectional study. Right? Dr. Sunny Sen Gupta.
That is a single point of time.
What?
So today, if you have a patient, tomorrow if you have a patient, and you just take one particular day, point in there.
Can it be like within 1 year I got these many cases and this is a cross-sectional study for 1 year? All are within same time within 1 year. Say bullseye maculopathy in HCQ toxicity. We get rare. It's very much well evident, but we get. So I tell that within a 5-year of period, number of patients having bullseye maculopathy documented on autofluorescence and OCT. Those patients.
Yeah. So if you include all the bullseye maculopathy in a particular time period, but only present. See the presentation. At presentation, what is their vision? At presentation, what is their what you call retinal finding? Then that is a cross-sectional study.
See, what you're reporting, what you're talking about is you're evaluating. For example, you looked at, say, 250 patients of bullseye maculopathy and in your clinic over the last 5 years. That's a retrospective case cohort which you're looking at. So it cannot be a cross-sectional study here, okay? So if you want, what you want to say is over a period of time, if you're looking at this sample of patients, say every 3 months, every 6 months, every 9 months, 3 monthly or 6 monthly, that becomes a longitudinal study. That's a prospective longitudinal study. A cross-sectional study, as Apurva said, your outcome and the event is at a single point of time. So you're just looking at a population of diabetic retinopathy at this particular point of period, and you're saying how many of them have a PTR, how many of them are having a CSME. So it's a single point study. It is not over a period of time, okay? You define the period and you say at this period, for example, a cross-sectional study can be population-based, can be institutional-based. A population-based study for a dry eye disease will be different from data which you would get on a institution-based dry eye study, because institution, everybody is going to refer a higher number of dry eye patients to an institution. Whereas if you look at a population, you might still be looking at the same morbidity, but your data might be different. Yeah, so that's something which you will have to understand. Yeah.
Please.
Will it be a cross-sectional study or is it a cross-sectional analytical
In cross-sectional, you can comment on prevalence of a disease.
Yeah, we are talking
if you're talking about incidence, yes, you have to, your cross-sectional study cannot talk about an incidence. So which means you're looking at newer onset of cases as well.
No.
Yeah.
What is the prevalence?
Yeah, so that's what. So you said incidence earlier. So I think you have to get your biostatistician on your planning protocol who will be able to define the statistics which needs to be applied for your data.
Right, but then it is the prevalence only. It's only prevalence.
Prevalence, it's a cross-sectional study.
So within how many, is there an upper limit as to what is the time period during which we should be collecting that data? That is what I am asking.
So that will depend on the study protocol you're defining.
Okay.
Yeah? That's how we are going to talk about your study protocol.
So it depends on the logistics also. If you can cover everybody within 3 months, then you can do it in 3 months. If you cannot cover it within 3 months, then 6 months. And from the same data, you said you want to compare it with another district where the radiation is, then it becomes a case control. So the same study can, when you do the
Or is
prevalence
it a
part,
cross-sectional analytical study?
No, so cross-section can't be analytical. Cross-sectional is always be descriptive. It's a one-time.
Okay.
Yes. So we'll go to the next
Yeah, thank you.
presentation. So, after Dr. Samrat has nicely given us a description of what is advanced search, I'm actually going to try and demonstrate to you, and then I'm going to show you that, well, all of this is perhaps things of the past, because there are some AI tools which I'll show you, and you can make a note and maybe bookmark in your laptops and use it later on, that you just have to ask it a simple question in simple English and not go through all this jhamela. That's somehow it looks like that a little bit. So, so the question I thought of before was a retina question, but now since we are talking about steroids and corneal ulcers, I thought we'll talk about voriconazole and use a voriconazole in corneal ulcers. So, this is how, so first and foremost, I am logged in here, and this is the PubMed website. And, it will be a little slow because the internet is a little slow. So, let's look at fungal corneal ulcer, right? And I'm just going to say add, and if you see that term automatically goes in this query box on its own, and let's say voriconazole, right? And I'm just going to say add, and when I click search, so these are mesh terms. So it says 228 results, showing results for fungal corneal ulcer and voriconazole, right? Now, instead of fungal corneal ulcer, if you say fungal keratitis and search, you will see you are getting 566 results. That's a huge difference, no? So fungal keratitis is actually the mesh term and not fungal corneal ulcer, right? So if we use mesh terms, we'll get a far better outcome compared to if we don't use mesh terms, which he was also saying. Now suppose we want to modify the search because say we are wanting to do a study on say intrastromal voriconazole. There's a lot of studies have happened on that. So how do we do that? So you come here and you just say this 566 search and click here and say add query. It automatically goes there. You don't have to retype all over again, and you just say intrastromal and add, and it is automatically come down, right? So this is the search builder which is happening. So the advanced search is, you're seeing the live demonstration of the advanced search, and when you click search, so now this is intrastromal voriconazole. Now you see there are 65 results, right? And the first study is from the RP Center group on fungal keratitis, a review of clinical presentations, treatment strategies and outcomes, right? So this is in the Journal of Ocular Surface. Next is combined intrastromal voriconazole and amphotericin B treatment for persistent fungal keratitis, right? So all of these has this intrastromal word in it, right? So, so that's how we have come down from 566 to, we are basically trying to refine our search. So think about using advanced search like a large Google map, where you say, I want to go to Yashobhumi, and it will start with the world map, and then it will zoom into India, then it will zoom into Delhi, then it will actually zoom into the point of interest. So we start with a relatively broad term, and then we start cutting down or chopping it down and saying this is where I want to go, or this is what I want to actually do. So advanced search really helps us in those. So it really depends upon your keywords, isn't it? So if your keywords are the right ones, and your keyword should be as close to the mesh terms as possible, though there is something called automated term mapping at the back end, where PubMed is trying to figure out, is this fungal corneal ulcer similar to something that I know? But like I showed you the example, it was, there is a very large difference between 200 and 500 terms. So this is how we come, into about 65 search terms. Now, how do we collate all of this information? You'll have to open all of these, right? At least the abstracts, and try to see what is given in that. So, the first one is a review article. So that really makes a lot of sense, where you will get a lot of information straight away from that article because it has reviewed a lot of those. But then, you still have to do that work. You still have to go into it. So, these are two AI tools I thought I'll show you. One is called Site, SCITE. And if you have laptops, you can actually open the Site website, and you'll have to log in. And, there is a lot of, whatever I'm going to show is almost entirely free. And so though I have a paid account, but then almost 80-90% of what you're going to see is free. So we are just going to say use of intrastromal voriconazole in fungal keratitis. Yeah. In AZOLE, right? Yeah. So these small things matter, so if you spell it wrong, it's not, so you are going to just ask it to do its own thing. If you see it's doing its own thing. The other very nice one is called SizeScape. So it calls itself the fastest research platform ever. And if you see, these are all the features which are available. So SciSpace is something also that you can look at. So you can do literature review, which we will look at. You can chat with a PDF. So you can actually upload a PDF. So let me show you some examples of what I have done. So so say maybe this is a paper which I had uploaded on somebody who, so this was, I was actually doing a review for non-ophthalmic, purposes. So you will have a PDF with you, you can upload it, and then, so you actually see the whole paper, and the paper basically is about, something else is Lewy phenotyping or whatever in blood donors, but you can actually type a question here, and it will give you the informations, so these are all the questions. Generate a summary for this paper. It just basically gives you this, right? So on a scale of 1 to 100, please rate this study of for novelty, methodology, and statistics, writing clarity, and prose, and it actually does a good job at that. You can create a podcast from that. If I'm feeling bored of reading. Now I want to actually get a 2-minute podcast on this. So you can actually create that, and it will take about 30 seconds and create a nice podcast in a Q&A format. So this is something which this website can do. We are now looking at literature review, right? So I'm just going to click that. Let's see what site has come up with. Okay, this already has done the work. So, I'll just blow it up a little bit. The use of intrastromal voriconazole in the management of fungal keratitis has gained significant attention due to its potential efficacy and safety. Voriconazole is a triazole, it goes through drugs about voriconazole. Then it talks about a series of cases, highlights the successful application of intrastromal voriconazole, etc., right? Overall, the combination of intrastromal and intracameral administration of voriconazole has also been explored. However, despite these advantages, some challenges remain. In conclusion, right? So, this is almost like a copy paste of an introduction for a manuscript. Not that you do it, but this could actually be, instead of staring at a white paper, these could actually help you in doing some of those things. But like Dr. Samrat said, you should be very careful of what this is doing, and you need to actually cross-check everything that it is throwing at you. So it's not just a copy-paste job, because you will get found out sometime or the other. And also remember that nowadays journals have AI, AI catchers, sort of. So most journals will realize that this is an AI written entire paragraph. So then, don't use it to write papers as such. But you could actually borrow ideas and the flow which is using, and maybe some of the references. But interestingly, what it also does is it creates this search strategy. Let me just show you that. The search strategy that was created by us. So it actually creates a search strategy. Search strategy here. So this is edit. So voriconazole efficacy fungal keratitis, treatment fungal keratitis voriconazole, intrastromal voriconazole fungal keratitis. And this is pretty much what we did in manually, right, in PubMed. And then we had to open and then get information. But it is actually simulating all of that and giving you a sort of concise information. So it is actually more helpful when you are sitting in your OPD, right? So so there is something which you are not really sure of, and you can ask questions, and you will get straight away answers from that. And here you can actually change these responses by going into settings and say I want reference from, or I want citation style of, say, Vancouver, right? And I want publications from 25 to actually 100. Let's keep it at 25. And when you say apply, so all of this actually gets converted into superscripts, right? So these are all superscripted here, right? So this is almost like a, it's a good write-up, but most of this will actually get caught in if you submit as is to a journal. But it could be a starting point, right? So this is site. This is, so let's see what this SciSpace does. I'm just gonna, let me just type this again. Intrastromal zone for fungal corneal ulcer, and this says deep review or high quality review or standard review. So let's keep it at standard for now, and let's say click, right? So it's actually going to ask you to refine your search. That's generally what it does. So there are questions I actually haven't explored. So I'm seeing the results as fresh as you are. So this is not rehearsed, because Dr. Samrat used the example of corneal ulcer. I'm using example of a corneal ulcer. So it says answers from top 5 papers. You could say from top 10 papers, and it's not going to change so much. So again, talks about something similar. Intrastromal has emerged as a promising treatment. Studies indicate this efficacy of intrastromal voriconazole, and it gives you some references, application in clinical cases, and comparative studies have been done. And this is the more important, or rather the more powerful part of this, where it talks about, so these are all summaries from these articles. So this is 10 papers it is showing. So first is talking about intrastromal voriconazole as a primary treatment in deep-seated fungal keratitis. These are insights from this paper alone, not a summary, but insight from this paper. And then you can create more. So if you say conclusions, it's going to create a whole for all of these. If you say results, it's going to create one more. So this is what you see so many times in systematic reviews. So where you get these kind of tables. So this is actually populating all of those, right? So this is from insights from this paper. This is conclusions from this paper. Intrastromal voriconazole shows better efficacy than natamycin. Further studies are needed for conclusive outcomes. And then it is actually populating the results for all. It takes a little bit of time. So these are AI tools which will help. These actually work on similar things like like we did with the PubMed, but this is all of course manual, right? So you put in these keywords and then you get articles, and then you have to open each one of them and read as much as you can, and then synthesize your own evidence. That of course takes time, but these will actually help you synthesize results much faster, and they could also potentially help you in writing your own papers later on. So there are plenty more. There is one called Elicit, which I am not showing you. That's so you could actually make a note of that. Elicit is actually even recommended by Cochrane now, right? So Elicit is recommended by Cochrane. So is this SciSpace, right? And interestingly, there are many, Site and Elicit, they are all actually headed by Indians. They are Indians in the Silicon Valley who have come up with some of these software, right? So these are two that you can definitely explore. And, there a lot of these are free for many things that you're looking at. And, so additionally, Site also actually integrates with, let me show you. So this is the same PubMed. One second, I'll just open that 65. Right, I'm just going to blow this up. If you see, it is integrating with my Chrome browser, and under each paper, I'm getting a box, which is not there in anyone else. I mean, you won't see this. So this says this is cited 121 times. Okay, and then so these are some of the metrics which actually gives, but this is the most important. This study has been cited 121 times. Right, so so Prof. Namrata Sharma is the first author of this paper, right? So so this is, if you want to cite something, this is perhaps one of the studies that you must cite, isn't it? So the next study is only cited 8 times. Maybe it's not such a great study. I don't even want to maybe open it and synthesize information from it. Remember, this is not an original article, right? This is a review article, so there is a chances of it getting cited more number of times. But the whole idea is to show you that site will integrate with PubMed and with many other websites and give you some of these metrics which really help you evaluate this. So this is only cited one time, and this is cited 86 times, right? So I am actually a co-author in this paper way back in 2015. This is cited 26 times, so this is 11 times. So this is how site integrates with your paper. And then the one at the bottom, this is the SciSpace, related papers and chat with paper. Let's see what it does. So once you click chat with paper, right? So this it actually opens up the paper, and then you can actually chat with the paper. This is actually getting ready to answer your questions. And again, if you see here, there is a site box, right? So even if you open that, the site box opens on the site, and you're getting the same metrics, cited 121 times. So can you summarize the paper? Please, you have to be gentle with the AI too, right? So summarize the paper, please. So let's see what it says. Overview of fungal keratitis from this paper. So this is all the things that the paper talks about, right? I mean, it's really giving you a lot of information, and it has access to the full text, though you are not able to see it. But these websites have tie-ups with all the publishers where the AI is able to crawl through all of these full texts, but we are not obviously able to see the full text. Right? So you will get a lot more information, looking at some of these, some of these summaries from the AI. So these are two things that I thought I'll show you about Site and about SciSpace, and I'm sure you are all ready to explore some of these technologies. And these are specific. Now we are going to look at domain-specific AIs, isn't it? So Perplexity is not domain specific, neither is ChatGPT, neither is some of the others, but then these are now domain specific AIs, right? So these are two things that I thought you could explore in terms of literature review. All right, so after literature review, we'll quickly jump into slightly different domain in terms of research. Ma'am, any comments?
I think that's a fantastic tip which you gave, and I'm still learning from you. Thanks so much for that. One doubt which I have is it that your Google search engine has integrated both PubMed and Site or Skyspace because you are a habitual user of these, or and you're a paid user? Because this usually doesn't come if the user is not using Skyspace or Site, right?
Yeah, so these
Yeah.
actually have Google Chrome extensions.
Okay,
Just
so
go
once
and
you
yes,
add it onto your
yes,
or
it's like
integrate it into your
yeah, like
extensions,
Zotero.
then it automatically
Yes.
okay. Yeah.
So it just
Yeah, then
integrates and it's free.
you
These
don't
are all
need to
free.
be a paid user.
No, no,
for
no. You don't
these,
have to be a
yes.
paid user
Okay,
at all, right?
gotcha.
So, yeah. No, so here if you see, when I looked at, when you look at PubMed, you can actually say best match. You could say most recent. You could say publication date. So here it does not matter on number of citations. So here it is best match, right? To answer your question, this is the manual one. Now, what this is based on is something that we don't know. It's something that the company is not going to tell us what this is based on, right? So this is top 10 papers, and a paid user can actually get even top 20. So I have logged into, so if you say high quality, you will get a little longer response. If you say deep review, you will get even more, deeper response, and you could actually bump it down up to 25 papers also, right? So, so deep review is available only to the paid user. So I have logged into this. So this is answer from top 10 papers if you see. So these are all like 3,000 a year, 4,000 a year. It's not, if you're really doing a lot of papers, even 3 or 4 papers a year, there's 3,000 or 4,000 rupees for the whole year. You could also do monthly subscriptions, which is like 400 rupees, 600. So site actually is 400 a month. 400, it's like dirt cheap, like really cheap, 400 a month. So I want to do my thesis, and I don't want to use it again. So I just take that 400 rupee subscription and get site to write my literature review in the long format with 100 references, and it'll really give you like a 3 or 4 page Word document, which is relatively long, and you can of course build on it. So so I don't want to give you more ideas with Vanati ma'am here, but generally you got the gist that you can actually get started with this.
Yeah, it's a useful tip. It's a
Yes.
very useful tip. But the point is, you'll have to use it constructively
Yes.
and not abuse it. So to learn how to interpret the help which AI is giving is not copy from it, but after you've already gone through your ROL well, it helped you to organize it, and then you can probably improvise and you can make it better. That's how you actually use it to help you rather than you do a plagiarism job on that.
Right.
That's how we should intelligently use it.
Yeah. So start it as it's like a primer. You can start with it, but don't depend on it. And trust me, AI also lies. Like, the language will be like so beautiful, you will never think about cross-checking, but it will lie, and then you'll say, "Arey bhai, this paper doesn't exist." They said, it'll say, "Oh, actually, is journal mein nahi, us journal mein tha." And then you'll say, "Okay, let me go and check that." And you'll say, "Oh, May issue 2022, there is no paper." And then you will ask it, "There is no paper." Then it'll say, "Oh, no, actually, I got this wrong and that wrong." And then after 4 or 5 attempts, it'll actually take you to that paper, and you will see that nothing about this is mentioned in this paper at all. And so these are called hallucinations. The more you use it, the more you'll realize that the AI actually could be lying. And so that will happen more with AIs which are not domain specific. So these domain specific AIs don't have those kind of problems because they are very refined to answer your particular question. Okay, so from sure ma'am, please, yeah. So now I'm going to take you a little bit into the statistics subject, but you will realize that again you don't need to know any of this. You can actually upload your Excel sheet to an AI that I'll show you. Just upload your Excel sheet in any form or manner. Kitna bhi bura hone do. So if you send a bad Excel sheet to a statistician, you will never get results. They'll say, "Oh, I'm this and that," and they will not understand the Excel sheet at all. But the AI will understand any kind of Excel sheet. And you have to just ask it questions. And what kind of questions to ask that AI is something that we are going to discuss now, right? So we'll spend about 15-20 minutes on some basic terminologies and then understand, how to write, because statistics is something that we don't understand much, right? So how to write that prompt for the AI to actually do your own, to do your statistics, right? So that's what we're going to learn. So I thought, that there'll be more people and I'll put up this slide, but I'll still ask you guys, how is the Josh? And I hope it's high, right? So there's not much time for these things. Let me just quickly go into the statistics. So there are two main types of variables. So I want you to concentrate on the left. I'm going to try to break it down and make it as simple as possible, so that just this basic knowledge will help you write prompts for your statistics software, right? So there are two main types of variables. One is continuous variable, the other is a categorical variable. Right, so continuous variable means something which is continuous in nature, and where it starts from the smallest value to the largest value in a particular, so we'll take examples and you'll realize. The other is called categorical. So categories, as the name suggests, categories or baskets. So we group people into baskets in terms of their common properties. So that variable or that question could have only two categories, say male and female, or success, yes or no. So that's like a binomial outcome. So that's on the bottom left. Then you could have more than two outcomes, like say response is fair, good, and bad, or something like that. So it could be more than two options. So if they are in an order, we call it ordinal. So simple example in ophthalmology is grades of non-proliferative diabetic retinopathy. So mild, moderate, severe, very severe. So if you have to categorize patients, you will categorize it as that. So there are more than two. They are in an order. So that is ordinal. And sometimes there are more than two options or more than two categories in that variable, and they don't have an order to that. Then we call that multinomial, and we generally don't use these. So continuous variable, also called as quantitative. So it takes any value between the lowest and the highest scale. So when you take ophthalmic examples, there is always a unit to this unit. So age is in years, could be in months, right? Could be in days for neonatal cases. BCVA is in logmar units. Intraocular pressure is in millimeters of mercury, right? Central foveal thickness or retinal nerve fiber layer thickness is in what is the unit of macular thickness? Microns, right? So all of these are examples of continuous variables. And categoricals, like I said, there are categories. Examples again is gender, male and female, success, yes, no. I told you some of these, right? Type of macular hole closures, type 1, type 2, type of trauma, either closed globe or open globe, right? Only two. So these are examples of categorical variables. And like I already told you, binomial is two categories, ordinal is more than two, and multinomial is more than two, but there is no order to it. This is one, you should look at this YouTube video if you can. You can again, remove your phones, maybe take a photograph or just scan it. You will understand how to make an excellent Excel sheet for your statistician. But like I said, a lot of this is getting outdated. So you need not really send your Excel to a statistician at all. You could make it as you want. But then there is a right and a wrong way of making an Excel sheet, and this will tell you. So when you have your your data in your Excel sheets, so basically you have each patient in a single row, or each eye in ophthalmology in a separate row, and columns are all the things that you have measured, right? You have measured age, you have measured gender, you have measured vision, and so many other things. So when you have all of this, first thing we do, or first thing you should actually do, is describe each variable. That is, each column in the Excel has to be described to the best of its ability. So we are like humanizing that column. So oh, this is my friend age, and then that age introduces himself. Oh, my name is age. My mean value is so and so. My standard deviation is so and so. So what do we use to describe that? And then so that's a continuous variable. You describe it in a different way. And then that variable could be a categorical variable. Oh, hello, my name is gender. There are only two types within me, that is male and female, things like that. So first and foremost, every column in the Excel should be described so that you understand, what is happening. So whenever there is a cricket match, the first thing we see is the batsman coming in and the bowler, and then they show their individual records, isn't it? They don't straight away start batting and bowling. So they first give you the description of that player, even if it's Rohit Sharma or Virat Kohli. They'll give you that definition of who this is and what they did, and then they will start interacting with each other. Similarly, in statistics, that's what we do. So how do you describe a continuous variable is in terms of mean with standard deviation. Now this is what we are going to ask the AI to do. If you have age as a column, you will ask the AI, please tell me its mean and standard deviation. instead of actually calculating, you will just tell the AI, tell me its mean and standard deviation, or you will tell the AI, tell me its median with interquartile range, which we'll discuss what it is. Then you will ask the AI, are there any outliers? Outliers are extreme values, right? And then show this continuous variable to me in a graphical manner. The best way to do is using something called box and whisker plots, which I'll show you. And the other type of variable is, of course, categorical variables. They are just percentages or proportions, right? So how many men, how many women? So there is 64% men and 36% women in this study, right? So that's relatively simple. Best way to show it is using bar diagrams. This is an example of a box and whisker plot. So instead of actually writing code, you can ask the AI, show me this box and whisker plot for intraocular pressure at different time points in the study. So, so this is pre-operative, this is at 1 month, this is at 6 weeks, and this is at last follow-up. Right, and this is all intraocular pressure in millimeters mercury. So this is a box and whisker plot. What it shows is, this box, and then these are whiskers. Whiskers are basically, the mustaches of the lion and tiger. So the central line is the median value. That means if you arrange it from the smallest to the highest, the one in the center is called the median value. The one at the bottom is the 25th percentile, the one at the top is the 75th percentile, and then this is the 90th and the 10th percentile. So basically you're telling the reader, or you're actually telling yourself, what is the distribution or what is the range roughly of all the values that are present in this IOP, and then these are outliers, that is extremely high values or extremely low values, which are very far away drawn from the median value. So here if you see preoperative intraocular pressure in this study was around 18 or 19, and then there are some people who had like 55, maybe 50 plus IOP, right? So what will happen if you drop these? The median will drop, isn't it? But the mean will drop, but the median doesn't change. So, we need to understand whether this is real or whether there is just a data entry problem or things like that. So instead of 26, somebody have just entered 56. So then that becomes an outlier that way. So it's important to understand how many outliers are there in each of your continuous variables, and your AI can help you with that. There's another example of an AI, which is relatively more complex. This is distribution of my running distances in every month for the year 2024. If you see, this is Jan. These are box and whisker plots. So if you see, this is there's not much variability at all. There is a little bit, So if you see this, distance actually starts going up and up and up from August to September, October, November, December. This is my own, so the median distance that I ran in the month of December at every run is about 11.5, 12 kilometers. Every time I went to run, I would run about 10 to 12 kilometers. That's like that's long runs, right? And then, I have done more than 21 kilometers twice in this month of December. So you can actually use some of this to your own advantage. You can use your own data, your blood pressure, your HbA1c, and some of these things, and get trends like this. This is an example from a DRCR network protocol which treats diabetic retinopathy using these 3 drugs: aflibercept, bevacizumab, and ranibizumab. That's Eylea, Avastin, and Lucentis, right? So, this is something which I have made from the data available from the protocol T, which is available on the internet to everybody. And we'll do this exercise live a little bit later. So, this is, if you see this is the mean thickness at baseline is 450-60 microns. At 8 weeks, that is after 2 months, the mean thickness in Avastin group is far higher, far higher compared to these others. So then, why would we actually use Avastin in other than the cost benefit maybe? So, using Avastin really for DME doesn't make a lot of sense because there's a huge gap between this. If you look at papers, this is also published in the publications, but we don't get that wow effect. But if you do your own analysis, you will never forget it because you did that own analysis, right? So, this is something that you can also do. Let's talk about analysis. There are 3 basic forms of analysis, and again, the AI will do it for us, but we need to understand. One is difference between variables, other is correlations, third is associations. Let's take some examples. What do you mean by difference? So when we have 2 groups, and it's a continuous variable we are dealing with, say age, or say macular thickness, right? So if there are 2 groups, say Avastin and Lucentis, what is the mean macular thickness of Avastin and what is the mean macular thickness of Lucentis groups at baseline, and are they significantly different from each other? Isn't it? That's an important question we want to ask. Or is that difference, what is the difference at 6 weeks or 8 weeks or 3 months or 4 months or whatever, right? Now, if there are two groups, and this is something that you now don't need to understand, but if you want to cross-check that AI, you need to understand that the AI should use the student t-test to find the difference. Remember, these are continuous variables. So let's simplify it and say intraocular pressure. Mean intraocular pressure at baseline in surgery A versus surgery B, right? So now it's a continuous variable because it's millimeters of mercury. And so two groups, if it's normally distributed, that means there are not many outliers and everything is near the mean. The standard deviation is not very large. Everything is sort of stacked up in the center. Then we use the student t-test. And a lot of the times it's not like that because there are outliers and the standard deviation would therefore be larger. That's when we use the Wilcoxon rank sum test. And trust me, even if you don't know any of this, it's totally fine. Before it wasn't, but now it is. And if there are more than two groups, like if it's aflibercept, Lucentis, and Avastin, now there are more than two groups. So here we use something called the ANOVA. ANOVA is the analysis of variance. And if you're trying to find differences between categorical variables, we use the chi-square test. Cat is chi. Categorical is chi-square. Let's take one example from a COVID study, which I had actually analyzed, and now this is published in the Lancet. So this is control group, and other group is remdesivir. There are 221 patients in the control group and 262 patients in the remdesivir group. Now the mean age in the control group is 57 years, plus or minus 12.9. Is the age a continuous or a categorical variable? Continuous. It's in years. So therefore we have mean and standard deviation, right? Remdesivir group, it is 58.8 plus or minus 12.3. So remdesivir group is about 1.5 years older, right? The mean age. When you look at the p-value, it is 0.15. What tests do we use to get this p-value? If you go back, it's a continuous variable. Student t-test, right? Simple, because if you see, whether it's normally distributed or not, for that you need to look at the standard deviation. If the standard deviation is large, how large is if it is more than half the mean? That's where we call it large. And if it is like that, then it is not normally distributed. Here it looks to be definitely normally distributed, right? So even if remdesivir group is 1.5 years older, it is not significantly older. So we do these tests, and the outcomes we get is p-values. Okay, now let's look at diabetes. 26% in the control group versus 41% in the remdesivir group had diabetes. This is what kind of variable? Yes, no. Continuous or categorical? Categorical, simple, right? It's a percentage. So what do we use here? Chi-square test. Cat is chi, that's all. So when we do this, it is a significant value. So there are significantly more diabetics in the remdesivir group compared to the control group, right? Now, if the death rate is more in the remdesivir group, will you blame remdesivir or will you blame diabetes, or we don't know about it, right? So, the remdesivir group definitely has slightly older patients, and many of whom are diabetic compared to the control group, right? So therefore, diabetes could be the reason why there are more deaths in the remdesivir group. So what I'm trying to sort of, bring into the context is the, relationship between the drug and the outcome, which is death. There are somebody who's poking their nose in between that, right? And that is diabetes. So what do we call this? Confounders, right? These are confounders. So when you do these head-to-head analysis, you will realize that, maybe there is something else other than the actual relationship, right? So, but direct head-to-head comparisons, when we do, we use these tests. Okay. And now, to find whether that confounder is actually influential or not, we do something called regression analysis. You may have heard about this terminology before. That means when the outcome is, that is whatever and that, the influencer variable, the main variable, the drug, say for example, is, so they are two linked to each other. There could be somebody who is pointing their noses in between. They could actually be increasing the effect, they could be decreasing the effect, right? And they could be modifying the effect in which ways we don't know still. So to neutralize the effect of all these, people who are poking their nose in between, to neutralize the effect of all that, we use regression analysis. So regression will actually tell you causality, right? So when people say, the COVID vaccine led to a lot of neurological deficits, or rather COVID led to more incidence of vein occlusions and arterial occlusions. We've all heard that, right? But when you look at very large data sets, you'll realize that that's perhaps not the case. Maybe there are anecdotal reports, but a lot of those patients who were reported from COVID time actually also had diabetes, had hypertension, had all these other comorbidities, right? So Which type of regression to use is actually again based on the type of outcome variable. It's a continuous variable used something called linear regression. And the best example of this is the SRK formula. You know, right, there are two types of formulae. One is regression formulas, others are theoretical formulas, right? Of course, theoretical formulas are more accurate, but the others are called regression formulas, right? That's where the word comes from, regression. Linear regression, SRK formula, where the outcome is IOL power, right? Is IOL power a continuous or a categorical variable? It's in diopters. Continuous variable. So, you know, that IOL power is a continuous variable, and those others are the influencer variables, right? So, whenever we're looking at an outcome which is a continuous variable, we use linear regression. When the outcome is a categorical variable, we use logistic regression, and the outcome is in terms of odds ratios. Dr. Apurva was showing us something about odds ratios, right? We don't have a lot of time to go into details of this, but you know, this is what generally regression analysis do. I'm going to quickly show you a QR code on an ebook which has almost all the information that if you want to actually scan it or something, you can get all, everything that I've talked about is there in this ebook. So if any of you want to take a look at that ebook, you could actually do that, or you can write to me and I can directly send it to you. Right, so this is about the basics of statistics that you need to know, right? So this is site, I showed you this about literature review. This is the size space. Let me open the AI software. This is called Julius AI. And this is not paid. Every day you can ask it 15 questions, but anything beyond that, it's going to then be charged, and it has a little bit of cost to it. Let me show you how you. So, you can again bookmark this if you want. It's called Julius AI. This is one of the best softwares there are. So what you really want to do is upload your Excel sheet. So this is that. Just click here, say upload file, and it takes you into the thing, and then you can upload your CSV file or Excel file, which I've actually already done. Let me show you that one. Workshop demo. Yeah. Okay. So because the speed was less, I have actually already written a few things down. So this is what I have done. Let me show you that Excel sheet from the DRCR protocol. So you can actually go to the DRCR network or the CAT trial and a lot of these NIH trials, and you will get the data straight away there. Data tables. Let me show you protocol T. So this is all the data there is. Let me just blow it up. So I have cleaned it up a little bit. So this is a patient ID. This is a randomized eye. This is the drug that they got. There is the macular thickness at baseline. And if I split it up a little more, this is baseline. They're at 4 weeks, 8 weeks, 12 weeks, 16 weeks, 20 weeks, 28 weeks, and then then I have truncated because it is there till almost 2 years data is there on that website. And then this is the ETDRS letters. This is not Snellens, but this is letters. This is at baseline. This is the higher the value, the better the vision, right? So 84 letters is 6/6 vision. So at 4 weeks, 8 weeks, 12 weeks, 16 weeks, this is pretty much what we need for our initial decision making. So I have uploaded this to Julius, right? So let me just show you. One second. Do you have the protocol T data set with you? I don't seem to have a data set specifically named as protocol T. So this is what I've done. I've uploaded that protocol T, right? And before we go here, there are two options. You can use Python and you can use R. There are two main softwares. There are other softwares. Most of you have heard about SPSS, right? SPSS can be used, but then you have to learn a lot of coding. The other is called Stata, which also has even more coding. And then the very sophisticated one is called SAS, SAS. That is even more coding. And the most complex one is called R. It is free. It is the best software there is, but it is complex to learn. It's very complex to learn because the coding that you have to do is, it's not easy. So what the software doesn't understand our language, right? So you have to actually tell the software what this is. So you have to write code. Let me show you some examples. So this is the code. This is our code. If you see, this is the Julius has written the code on its own. That's what it does basically. It converts your query in simple English to R code. And then it runs it on R because it's free, and then actually spits it all out, right? So this is the one, so then let me see. Now this is what it says. I've analyzed a protocol T dataset which contains clinical trial data comparing 3 different drugs for treating diabetic macular edema. The dataset includes 660 patients, 3 drugs, aflibercept, bevacizumab, and ranibizumab. Measurements of retinal thickness and visual acuity scores are present. So these are 3 numbers: 224, 218, and 218. Mean retinal thickness over time by drug. So I mean, I had told it to do, so this is what it has done, right? So I showed you this, right, when I was making that presentation, right? So this is what you can do. Mean ETDRS score over time by drug. So again, if you see aflibercept, so this is the mean and this is the standard error at each time, right? So if there is an overlap of the standard error, that means it's not significantly different. If there is no overlap, it is significantly different. So if you see aflibercept, it's in orange, is far above, right? So the vision in aflibercept group is better than the other groups. And of course, again, Avastin is lacking far lower. So again, if you see, Avastin doesn't really cut it so much, where, the visual, the thickness remains around 350 and more than 350 throughout. It's not really drying as such. This is, reduction, or how much it dropped by, minus 50, minus 100, and minus 150. Again, if you see over time, aflibercept has significantly more drop in macular thickness. And, so this is all the code. It just writes code on its own, right? And then, the question I asked it was, what are the percentage of patients with retinal thickness less than 300 at 4 weeks? That is after 1 injection, right? So in the aflibercept group, this is the aflibercept and ranibizumab group, there are 28% patients who had thickness of less than 300, where we don't want to say re-inject or so. And that means these are the patients who might do well without a loading dose, right? So it's almost a third, no? 28-30% is like a relatively large number. So then people who are getting treated with aflibercept and ranibizumab may not actually need a loading dose as a blind therapy that all of us actually, a lot of retina specialists do recommend. This is true for DME. It is not true for AMD. It is not true for RVO. I have done all the analysis from those trials, CAT studies and all. It doesn't work, but in AMD it definitely works. So you can actually do a PRN from baseline. And so these are all very specific questions. So the last question I asked it was, so this is, it actually is giving you some other metrics. 1 second, yeah. So this is reduction in category. That is, there is an increase at 4 weeks, 0 to about 10% reduction, 10 to 19% reduction, 20 to 29, 30 to 39, more than 40% reduction. When you look at the Avastin group, this is bevacizumab, right? So when you look at this increase, it's quite a large number of people are actually having increase, and about, when you look at increase and 10% change, that's like very minimal change. It's almost close to 50%, right? So there is increment also. If you give injections, it could mean that it's not responsive. So this number is almost 15% in the Avastin group. 15% don't respond at all, right? And so this is only after 1 injection, of course. But overall, if you see in Avastin, it either doesn't respond or it responds to about 10% drop from baseline, whereas it's far more. And if it 40%, this is all basically decision making in clinics, right? So anybody has a question on this data set, we could actually ask and see how it works. Anybody who does retina and wants to ask a particular question to DRCR protocol T, we could ask any particular question. Anybody? Let's look at vision. So how many had greater than or equal to plus 10 letter gain? Yeah, you'll see now. Yeah, you have to write some code. So I'm writing that code below. I'm not writing code, I'm just writing the prompt. Yeah, you have to ask. So we'll see how many had. That's what, so I'm going to show you that. So for, the question, so what I did is with macular thickness, let's do the same thing with vision, right? Again, is a continuous variable. How many had more than or equal to plus 10 letter gain at 4 weeks compared to baseline? So that's a question. It says thinking. And if I can blow this up a little bit. It's a little difficult to blow it up. Once again. Yeah. Okay, let me analyze the ETDRS letter score changes from baseline to 4 weeks. Now it's going to write code. It's a little slow. The internet is a little slow. So you will see the code coming up on its own. Hopefully it actually works. Go ahead. See, the code has come up. Yeah.
There's something called as R commander. You know R?
Yeah. So basically,
So there's R commander. If you use that package, it comes like a normal this thing SPSS thing, and that is very easy. You don't need to learn the code then.
correct. So
You have to just download that package, and it's completely free.
right.
So this thing size is not a problem, because you can have any size of data set.
Right.
R doesn't have any limit on size, I think. So R commander is a fantastic
Yeah. So
free thing.
correct. So R has an interface which we'll call R Studio, and then R Commander is a package which has many things, but doesn't have a lot of things. I mean, R works on something called packages. Where there is a group of commands which have been loaded. So R command is something which works on, suppose you want to do some complex analysis, then that doesn't work. So then they are all coming up now, but this is actually, it does a lot of things, relatively simple things it does. Yeah, it does that. So, so this is the question we asked. So this is gain. Oops. So I'm just going to try and reduce the size of this. Okay, to see this well. So this is the gain category, this is the loss, right? So if you see loss, this is percentages, very similar loss. If you see gains, more than 15 letter gains is what, it's really good, but more than 10 letter gains is this one. So, this graph has been made by the software itself straight away, right? So this is aflibercept, this is bevacizumab, this is ranibizumab, right? So 10 letter gain, 10 and more, so 10 and 15. So these are the ones which have 10 or 15 letter gain. Now if you see bevacizumab and ranibizumab are doing similar. This is at 1 week. At 4 weeks, that is after one injection. But aflibercept is again little more than that, right? So more than 25% have gone. So, so this is something that you can do. Last thing I'll show you is, show me a line graph. Comparing vision at 4 weeks in the 3 groups with standard error bars, right? So this is the question I have put to it. So it's thinking, and then it will come up with a graph similar to what you saw for the macular thickness. Yeah. So you can go to the DRCR net website, and you will see protocols, and then you will see one thing on data. I mean, there is data on, so all protocol I, protocol T, all of these are now publicly available data sets. You can go and you can download those, and then you have to merge. Yeah, you have to do a little bit, but you can use it then. So this is how it is showing. This is at 4 weeks. 1 second, let me just.
Sir, if you want to ask any, want to write any questions, before that we have to upload the paper in this software? Or
Yeah, you have to upload the Excel sheet, the data sheet. Yeah, so if you look at this bottom, this is you say upload file,
okay,
and then you upload your Excel sheet, and then you start asking questions. So at 4 weeks, the the vision in the aflibercept group is much higher compared to the bevacizumab and ranibizumab groups are doing pretty similar. But so that is slightly lower vision, but the aflibercept group definitely has again better vision, right? So the macular thickness reduction is also far better in aflibercept group, and vision is definitely, this is only after one injection, right? So, so these are some of the things you can do, and it will show you. So you can ask for a particular graph, and then you can actually say at 4 weeks, 8 weeks, 12 weeks, and it will show you that also, right? So this is something that you can use as well. 15 questions per day is free. After that, there are paid versions. So, initially you will see that most of your prompts are not coming up with the right answers. So maybe you, after asking 15 questions, maybe 1 or 2 of your queries are getting answered. But then as you become better, your prompts start getting better. And that is when, you will utilize all those 15 on a daily basis, and you will get good answers. And again, annual payment is about 30,000 or so. That is an annual payment, but monthly payment is about 3,500 or something like that. So if somebody is doing one study and wants to get some analysis, if you send it to a statistician anyway, you're going to pay more than that. And then, whatever comes back is sometimes difficult to interpret. So then you can actually get this for a month, do your basic statistics for a week, 10 days or whatever, and then say, I don't want to renew. So Julius is something that we can really use, and do your own statistics. So understand that basic concepts. What is this continuous? Is this categorical? Do I want to do direct comparisons? Do I want to do regressions? What is the kind of graphs I want to make? So this is just a primer. So Julius is the website that, the AI, which will really help you do your own statistics. And, there will of course still be a learning curve, but, it is far better than actually trying to code in R or trying to code in SAS. So, this is something that I strongly recommend, and you can take a look at that. Yeah, so if the paid version has actually 10 GB space for you, 10 GB. And if you don't do anything, it will still stay for a month. And then, it will tell you that, oh, ye dormant ho gaya, I'm deleting it. And then it deletes on its own. So, but then 1 month is long time, yeah. No. It will stay for a month. This thread, this thread will remain active. There are all these threads, right? So all these analysis that I've been doing. So all these threads will remain active for a whole month. Yeah. And then, yeah. No, any Excel is fine. CSV or Excel, either is fine. Yes. Excel or CSV, any is. Yes. But this takes Excel also. Yeah. Okay, so with this we come to the end of the statistics module, and we'll go to the paper writing module, and I'll just disconnect it. Yes, please go ahead. Yes. Yeah. So before that, I'll invite Dr. Akshay to come up and set his presentation. Till that time, I'll answer your question. So yeah, so basically you can upload in ChatGPT also, and then it will also give you something similar, but it will only work on Python. At the back end you won't see what coding is happening. It will only work on Python, and the images don't look as good as the R one, and you actually can't cross-check that. So it will only work on Python, and many times there is hallucinations. So it is not a domain. Like I said, no, we now are going towards domain-specific AIs. So if you want to do analysis, we want to use an AI which is specific. So it will give us, it will give us option of R as well as Python. So to answer your question, we need to write that the statistics has been done with R, and the the latest version. So currently it is 4.3.3, and then you can write using the Julius AI interface. That's pretty much what we have done, right? We have not written the code ourselves. We have used the AI to write code, but actual analysis is done still using R only. Or if you're using Python, if you're drop down and using Python, you have to then write Python, the latest version or whatever that is. And now it is, I think, some 12.3 or something. And then say using the Julius AI interface. Yeah, so then that's pretty good enough. And, most people who now understand these things, they realize that, the analysis, the backbone of the analysis is the same as before. Only thing is that, manually writing that code in R is quite challenging. It's quite difficult. And you are sure to get bottled up, even if you're using some packages which have a lot of things going on. Say you want to do some survival analysis, or you want to do, there are other complex kind of analysis which you will get bottled up with using. And learning R coding will be the best, of course. If you can spend time, sit, break your heads and learn to code in R, then you can actually develop chatbots. You can develop whatever you want. But then that is quite complex to do. Therefore, learning R is almost not an option. I've spent almost 2 years trying to learn R, and then I gave up, and then I said I can't use R. But now, we have this magic, which is an interface between, and we ask it questions, it posts it to R and sends us the reply back. So that's pretty much it. So we'll go ahead, and I'll invite Dr. Akshay Nair, a very good friend of mine, to talk about writing. We are now graduating onto actually writing and passing on our information, to the readership, to the journal. and he's going to start with writing case reports. He's a very accomplished oculoplastic surgeon in Mumbai. He is, there are a lot of things to read about him. He did his oculoplasty, fellowship at LV Prasad, and he's part of the Stanford University 2% list of global researchers and many more things. So I invite Dr. Akshay to talk to us about case reports.
Thanks. Thank you, Dr. Sabyasachi, for the invitation. I think as a beginner, I've targeted my talk as being for people who are starting off writing, or who've probably not yet written case reports, although there are very few who probably have written original articles and not case reports. But I do think there is a place for case reports. They are still one of the blocks that go in building your entire spectrum of what all you can write. Because at the end of the day, even if there's a case report and you think it's a low-hanging fruit that you don't want to pluck, not publishing that is a crime, because unpublished data is equal to no data at all. You have to publish because that is the reward for your labor. It's your place in science that is unalienable, and it eventually build your reputation. Don't sit on it, because nothing becomes outdated faster than science. So this is my overview of. I'll talk about the power of a case report. Of an interesting anecdote, an interaction. How do you structure a case report? What can be case reports? How can it be reported? The basic principles of writing it, and where can you publish it? So, there have been some historical facts in medicine that have, things that have changed the course of history in terms of how we practice medicine, all of which have come by through small case reports. This is a single case report. I mean, not even a case report. This is just a by the way note written to the editor of the Lancet in 1961, where this gentleman called Dr. McBride, who's a GP in Australia, says, "I am seeing babies born to mothers who are taking thalidomide with some kind of abnormalities. Have any of your readers also seen something like this?" And that blew the lid off of one of the biggest associations or causalities between phocomelia and thalidomide, and how they did an investigative piece and found out that the manufacturers were aware of this and they deliberately misled the medical fraternity. Another something similar, how they, in 1981, they noticed how Kaposi sarcoma was common amongst homosexual men, but it was only years later that they could put the two together, that it is actually related to HIV. Similarly, this is an eyelid lesion, a common thing that we know. It's a capillary hemangioma. We give, now the gold standard is no longer intralesional injection of steroid, it is oral propranolol. And multiple reports have come about. But how it happened is because steroids were still the first choice of treatment for hemangiomas, and there was this child with a huge hemangioma who was being treated with a lot of steroids for that. As a result of the steroid overdose, he developed obstructive cardiomyopathy, and to relieve that, he was put on propranolol, and what they saw was, without the steroids and only the propranolol, that lesion resolved completely. This paper has then gone on to be cited more than 4,000 times in the past 10 years, and that is the power of a case report. A lot of times, people tell, people in practice say, "I've been in practice for 18 years, why should I bother writing? I'm happy doing what I'm doing. What do I get by writing a paper? It's not for me." So this is an interesting case. A 58-year-old patient, female, had a right-sided lump in the right-sided breast lump and was excised by the surgeon, sent to the pathologist. You can see that there is a caseating granuloma in the center, surrounded by a cluster of tumor cells. So it was a malignancy with a tuberculoma. It was interesting, and the pathologist told me, I found this interesting case. I asked, "Okay, how rare is that?" And so I said, "I don't know, check with PubMed." And the pathologist said, "Who is PubMed?" So I said, "Okay, well, I'll help you." And then coexistent tuberculosis and breast carcinoma, not much reported. So I said, "Wow, then I think you should write it up." The pathologist asked me, "But where do I write it up?" I said, "For a journal." "Who's going to read this journal?" And so, this is how even the uninitiated can be initiated into writing. This pathologist was 66 years old at that time and in private practice for 38 years. She did eventually go and write it up. The coexistence of a carcinoma and tuberculosis as a case report was published in an index journal. So her first case report was when she was 62, and now she's gone on and over the past 6 years written 14 more peer-reviewed papers. She happens to be my mother. So if someone at the age of 66 can start writing, I don't think why people should not start writing at any time. So why do you publish? Because it undergoes peer review and it establishes that what you've done for that patient is the standard of care. It improves visibility. It establishes something new. And like I said, it's a permanent place in science. And if it's a rare thing, there are going to be people who will stumble across a similar complex case, a rare case, and this will always help them. I understand in the pyramid of the evidence, the quality of evidence, this is unfiltered information at the base where you have case control studies or case series, but you can always give it a twist in the form of a photo essay or a clinical challenge. So my next thing that I'll talk about is what can be a case report. So I've put this, clumped this into 8 different types of presentations that can be tweaked or spun into a case report. Could be a single case or a short series. One is if you have a unique presentation or a unique surgery or a unique treatment for a common condition. For example, now dacryocystitis is not uncommon, it's very common. But what was unique in this case was that the reason for the dacryocystitis was a lump in the lacrimal sac, which was from a metastatic hepatocellular carcinoma. So this is a unique presentation of a common condition, which could definitely qualify as a case report worth publishing. Or this child was sent to me for a suspected conjunctival melanoma, and what it turned out to be Bitot spots, but because the child was being applied kajal every day, which was a homemade kajal, those particles and the debris would keep accumulating over the rough surface of that Bitot spots and give it a black appearance. So again, a rare presentation of a common condition. Unique imaging or pathology or diagnostic findings of a common condition. So, for example, this was published in Ophthalmology. Now, this is a retinal astrocytic hematoma, not uncommon. They've done a wide field fundus photo. They've done on-fast OCT infrared scans. They've done an angiographic flow, an on-fast angiography, and demonstrating the plexus of the capillary vessels, which gives you a very nice multimodal view of the same lesion from different investigative modalities. So, again, unique imaging of a common condition. Again, subretinal drusenoid deposits. But what have they done here? They've done color SLO with a retro mode imaging. Again, a common finding, but a rare imaging modality. A simple condition, infiltrative breast carcinoma, which had metastasized to the optic nerve, but a classical imaging finding, which is the tram-tack appearance. So, a unique or a hallmark finding, but seen very classically of a common condition, again, can be there. A rare adverse reaction that you're documenting that with established causality. You just can't throw it in the air saying, I think this was what was causing it. As long as you have enough evidence to say that, yes, there is causality, you can do that. A drug causing myopia. Aripiprazole is an antipsychotic. The patient was put on antipsychotic 3 days later, said, I can't read. It turned out it was aripiprazole-induced myopia, where a child who developed reduced vision was being treated with immunoglobulin for Kawasaki disease, and this was keratopathy due to IVIG. And as soon as the IVIG was withdrawn, the keratopathy regressed. So again, with evidence to establish causality, a rare adverse reaction can be a case report. A previously undescribed association that you think you've stumbled upon, or you're just hypothesizing that could be an association. Something that we saw a lot about 4-5 years ago, mucormycosis associated with COVID. So this was the first paper that put it out in late 2019, in early 2020, saying that, hey, we've seen a few patients, all of them have severe COVID, and all of them have mucormycosis. Could there be an association? And this was the first case report that came up. And yes, as it turned out, there is an association. So previously undescribed association of an old disease. Could be a new disease or a new manifestation of a disease that's been just stumbled upon. Similarly, in COVID, we had a lot of things that could present as the first sign of COVID, like, for example, conjunctivitis, or some patients had clots, like thrombosis, like Dr. Sabyasachi mentioned. So, a new finding of a new disease or a new finding of an old disease. On an extraordinary documentation of a common condition. I know this also falls in the unique imaging or diagnostics of a common condition, but again, multimodal imaging of a pigmented vitreous cyst, you've taken it from different angles. Or a rare or a unusual complication or a treatment failure. Not all good outcomes only have to be published. Sometimes there could be a complication also which could be published, and you let people know that this also can happen. Something as simple as intrastromal cannula injury in cataract surgery. How often have we had that, where you're trying to do you inject either visco or hydro and you don't realize that your cannula is blocked, and as a result of that injury, pressure, you end up causing intrastromal. So what they did OCT, they did good high quality images, clumped together a case series or case report, and then a series documenting it. So it's it's not you don't need to think only for unusual and rare things. But how do you begin? You first start subscribing to journal TOCs. Every month, whenever the journal gets released, it'll come to you. You don't need to actually pull out and read every article. You could just skim through and see, hey, what are the papers of relevance that are being published? What are the case reports that are coming up? And something will click, hey, I had seen something similar. Maybe I should write that, or I could write something similar like that. Take your time to read, because only when you read can you then start writing. If you're focusing on a subspecialty, then pick out subspecialty journals. Collaboration is key, because sometimes you may have all your images, but you may not have the pathologist's input, you may not have the radiologist's input, you may not have your cornea colleague's input or your retina surgeon's input. So collect all of it, collaborate. Remember, predatory journals are there. The number of predatory journals for case reports is very high. I am not for one to, who would be in favor of these predatory journals, because a lot of them are not even indexed and they charge a high amount of fee. So to write a good case report, you need documentation of all your images. I think good images and good diagnostic investigations are the backbone of a good case report. Always examine the eye, the patient and not the eye only. That way you'll be able to get the associations that are there. So once you've got all the data, go through select your journal that you want, see the guidelines, and then prepare your manuscript based on that. Condense and compartmentalize. Now, of course, I think Dr. Sunny will also mention about how AI can also help you trim and polish your manuscript. One thing that I always tell my residents and trainees also is, whenever you're writing a case report, in your discussion, think of few things. First, highlight why your case report is unique. Second, compare your case report with previously published case reports. And then give a summary of why you thought it is worth publishing, in terms of what is your message that you give. Which is where this table that compares previously published similar case reports is always important. Try to add references that are more relevant, and avoid obsolete Duke Elder references, like I call them. There are a lot of journals that are still accepting case reports, so have to be patient. No double dipping. Don't try to submit the manuscript to 3, 4, 5 journals at the same time. You'll be surprised. Sometimes the reviewers are almost always the same for popular journals. Sometimes you'll get a revision. Be to the point. Thank the reviewers. If you agree with them, great. If you don't not, then be polite and be clear and firm on why you do not want to go ahead with this revision with good reason. Do not give up. There is this, and I love this paper because it was a very unusual paper that we published about, which we thought was unusual. It was a patient who presented with a chalazion-like lump. We curated it and thinking it's a simple chalazion. On histopathology, turned out to be isolated amyloidosis, and we actually worked up the patient systemically. Nothing. It was just a unilateral primary cutaneous amyloidosis. We thought it was rare enough. It was rejected by 17 journals. This was in my second year of residency, and in my fourth year of private practice, after 7 years, through 17 journals, it got published. So I always talk about this paper as a paper that pushed through multiple battles and 17 journals. So there are, 17, which means there must be at least 17 journals that accept case reports. Yes, whether it's specialty, whether it's general ophthalmology, a lot of them accept case reports. All of these journals that I've shown here accept case reports. Some other than these can also accept case reports in the way of pictures and perspectives. You can put a clinical case challenge, images in medicine, like in NEJM, a patient with microspherophakia, undilated, dilated, and then post-surgery. You can just put a nice, this was done by Dr. Annie Mathai and Anil Mandal and LV Prasad, was accepted as images in clinical medicine. Similarly, congenital sutural cataract, just a high quality image from PGI Chandigarh, made it to NEJM. So you can also think out of the box. Other specialties like tropical medicine, microbiology. This was endogenous endophthalmitis causing cellulitis in a pregnant patient by MSSA, and we published in a microbiology journal. So think out of the box. You can also think in allied branches like ENT, pathology, microbiology, etc. So to summarize, remember whenever you publish for case reports, rejection is usually the norm. So you always get to learn something, revise it, refine it, and resubmit it. The devil is in the details, because like I said, imaging and documentation, high quality images will always get you that extra X factor that convinces the reviewers to publish it. Keep a cache of all these interesting cases. You never know what can be published. This was a patient case that I had operated, and I had sent to Dr. Hunawar asking for, so this is what I did. What do you think I can do? I said, first thing is you give me the high quality image. I want to put it on the cover. So you never know what can get published. This was a Merkel cell carcinoma of the upper lid. So always document and keep these cases close. Take the time out and take the effort out to put these two. Case reports are the lowest hanging fruit, but they're always rewarding like any other paper. Thank you.
Thank you so much, Akshay, for that very, very illuminating talks, right from Focomelia to, to current times and your own papers and your mother's experience. So thank you so much. And any questions you have for Akshay, we can take them now, and I'll till that time set up my own. How many of you have actually written case reports, or at least tried to write case reports? You could, because it is a low-hanging fruit. And so a lot of journals actually talk about case reports, but don't call it case report. They call it pictures and perspectives. That's what ophthalmology calls it. Ophthalmology, which is like the top tier journal with an impact factor of nearing 8 now. They call it pictures and perspectives. JAMA Ophthalmology, which is an impact of more than 7. It doesn't call it case reports. Anyone knows what it's called? They call it ophthalmic images, right? Yeah, they have this section called "What's your diagnosis?" And it's something which there are differentials and other things. So you have to present the scenario, and then it'll, the reader will get through it like a quiz kind of. So all of these journals actually do take case reports, but then they don't call it case reports. So that's about it. So and I'll show you some online softwares which you can use to see which journal you could actually submit your case reports to. Thanks, Akshay. Yeah, thanks so much. So the last thing we'll talk about now is actually writing original articles. And so, like we've been discussing all of this, right? So where does the idea come from? And that's something that I'll show you in one slide. But we've talked about the study design, and what is the best study design for my study is what you must be interested in. So for that, you need to understand what are the study designs and which out of these will answer my study question the best and is still feasible for me, right? So Dr. Apurva talked about those, and has anyone done this before or not? Something that Dr. Samrat told us in terms of, and we saw some live examples of, how to do a literature review. Then we talked about statistics and, how you can actually do your own statistics using AI. Now we are talking about writing and publishing, where we talked about case reports. Remember that any topic that you choose has to be novel, that is, hasn't been done too many times before, has to be doable in your own settings, and then of course has to be publishable. This is how you can get to choose some good research topics, right? So, it's well and good saying, this is a eureka moment and I want to do this. But then, is there a systematic way of actually coming up with consistently coming up with research topics. So these are some ways you can do it, like, many people have mentioned before, subscribe to the table of contents of journals. So, so about 5 or 6 top journals in your area of interest, say cornea or retina or glaucoma, and maybe some common journals like ophthalmology, JAMA, American Journal, and things like that. So every journal will publish about 10 original articles every month. So if you are subscribed to the 5 journals, then you are getting 50 articles in your email inbox, at least the headlines, right? You are getting the title and you are getting a link to go to the abstract. And then if you read 50 titles, don't you think you will be interested in at least one of them, right? I mean, the whole intention has to be, can I do this? So out of 50, maybe you could do one, but still in a year you will generate 12 good ideas, right? So that's quite a large number. So subscribe to e-table of contents. Other is PubMed updates. Like we saw some examples of how to do a literature review. What you can actually do is tell, if I have time, let me just try and show you how to do that. So you go to PubMed and let's go to the voriconazole example. And so if I go to advanced search, you will see the previous. And so this is the 65 search that we came up with, right? Intrastromal voriconazole for fungal keratitis. The 65 results, right? Now what you can do here is, let me just blow this up a little bit. You can go, this will only happen if you are logged in. So I am logged in here, and you say create alert, right? And you click that. Now say this is something that you are interested in, you want to do this in your practice, in your institution, or you are doing a thesis on this or something. You want to be updated on what's new in this, right? So this is all which is getting the name of the search items. Would you like email updates of new search results? You say yes, of course, that's why I'm here. You say frequency, you can say weekly or monthly or daily. Let's say monthly because it's already there, or let's say weekly. And then it's saying which day do you want it. Let's say maybe Saturday evening I have some time, so let me do it on Saturday. And this is all the same summary, and then maybe 5 items. You could then do 100 items, which is very unlikely. Let's leave it at 5, and you say save. So your new search has been saved. Edit your search settings, and you can actually go there and see that that actually has been created. So what this will do is every Saturday evening, whatever comes on this topic, you can actually change it, right? Would you like email updates? Maybe weekly, say you make it maybe monthly, or you can actually change it, right? You want a summary or you want abstract in your email inbox. So let's say save. So now every month, on the first Saturday of every month, I'm going to get an update on what is new in intrastomal fungal keratitis, right? So these are all ideas for future studies, isn't it? So the where we started off with this PubMed updates, so you can actually so e-table of contents is not in your control, because whatever is getting published is getting published, and you need to then think about, can I do this? But PubMed update is something that you are telling PubMed that I am interested in this, let me know what is new, and then you can actually see what is new, and then you could perhaps do that. So that is another way of getting ideas. Ideas from clinics is also a really important way of doing this, right? Where don't stop asking questions. We began with that. So ask meaningful questions. If you ask enough questions, that is when you will actually get the right question. So if you ask maybe 10 questions, 1 or 2 may not have been answered at all. You can join collaborations. There are organizations like AIOS and many others who actually get people together and do some collaborative work. Journal clubs are a really good way of doing, getting fresh ideas, where you actually go through a paper as a group, you critique that paper, and you get a lot of information on where that was done well, whether the statistics are done well, and others. And then at the end, every good paper ends with what is new that can be done, or future trends or future studies could be done on something like this. So they are also ending with most top journals like JAMA or ophthalmology papers will end with something that can be done in the future, right? So reading full text and having journal clubs really helps. Of course, going to meetings like this, and going through to sitting in free paper sessions, and maybe attending the posters that have been displayed will also give you some ideas. So these are some examples of getting good research topic ideas, right? Something which has not really been done enough times in the past. These are e-table of contents already been talked about. Now we are talking about writing, right? So we've looked at how to get an idea, how to design a study, how to analyze that paper, how to do a literature review using AI, how to do analysis using AI, and I've given you all those tools. And now we are actually looking at writing the paper, right? So we are really giving shape to all of that you have done. So your ideas need to now come in paper and then get submitted to a journal, right? So it really follows the IMRAD format. I think most of you would know this, right? So I stands for introduction, M stands for method section, R stands for results, D stands for discussion. So IMRAD is the format which is to be followed for any original article. So this is what I've talked about. So before going into the nitty-gritties of each, you should remember that your document is never a final document. You need to write a first document and then circulate to your co-authors, refine it, write another second draft, then maybe circulate it, and then fourth, fifth, sixth, maybe the final draft. So I've always seen somebody will write and then they will label it as final. This manuscript _ final, and then it becomes like final 1, and then it becomes like final 2, and then you're like fed up. It's like, I don't know, and then you will end up submitting the wrong version to the journal, right? So it's always a good idea, therefore, to write as V1 or version 1, and then if the changes are happening, V2, V3, V4, that means the one in your folder which is at the bottom most is the actual final version, right? And still you don't call it final because it is going to get reviewed. You're going to make changes in that as well. So so it's never a final document. It's always a work in progress, like like most of us are. So a little caveat before we go into the nitty-gritties. So how do you write an introduction? So when an editor is looking at your paper, what is it that they will look at for sending it to a review, or straight away reject it outright without even a review? Has it happened to any of you where the paper has not even been reviewed and just rejected straight out? And that does happen quite a bit, especially in the top tier journals, right? So a lot of it has to do with the introduction, and that it has not been written well. So the introduction really means, like I gave you the example where you want to zoom down into New Delhi. So it's like a Google Maps where you look at the whole planet, then you zoom into India, and then you zoom into Delhi, and then maybe into Yashobhumi. So this is perhaps not playing enough. So so the whole goal has to narrow the reader's attention to your topic of interest, right? So it has to give a concise review of literature, that is what is known or what has been done, and very importantly, highlight the lacuna or the gap of what has not been done. And why is it important to fill that gap? Right? That's how your introduction should go. So initially you talk about some larger things, so examples is, so example is, I think I have an example. Yeah, I'll show you the example. So generally the idea is to say what is known, more importantly what is not known, and therefore that is where a lot of people will falter, where you will just say what is known and then you'll straight away go to the method section. But then what is not known? This is not enough for me to send it to a review at all. So remember, some of the top tier journals, including IGO, the editor will see like 50 papers a week. Like 50 papers a week. It's like 200 papers a month. So how many in a year? 200 papers a month is like, do the math. 2,000 papers a year. An editor will typically go through 2,000 papers a year. Do you think they will go through entire nitty-gritty of what you've written? No. I have been in that position. What I would actually do is look at the abstract. If it makes a little bit of sense, I would go to the end of the introduction and see if the author has actually given that gap or the lacuna. If that is not there, then nothing doing, not even going forwards, because there are too many submissions to process. And then people say, "Arey, humne itna kaam kiya, itna analysis kiya," but you did not tell what has not been done. Perhaps you haven't even done that literature review to see that it has not been done, and maybe the reviewer will do, and maybe the editor has also done a literature review and found that, oh, already there are some 7-8 studies there, and you have not even cited any of those in your introduction. So that introduction is not well written, and therefore it just goes for a toss. So so what is that lacuna now? What is the gap? Meaningful gap for a journal is very few studies are there to address this question, right? That would obviously be the best gap that kisi ne aur nahi kiya hai. It's never been done, so I'm doing the first time. That obviously is the best one, but it's not always possible for us to do such novel research, right? So sometimes you'll say, previous studies have been done, but sample size is small, and therefore we are doing a much larger sample size, and that is feasible in our context because we do have large number of patients. You could also say so and so aspect of disease has been studied, but something else has not been studied. Many aspects of disease, so risk factors, clinical features, treatments, outcomes, factors affecting outcomes. There are so many different things which you could actually base your study on as a primary outcome. Therefore you could say outcomes have reported, but risk factors predicting outcome has not been reported. Let's say, for example, endophthalmitis incidence has been reported, but what are the risk factors for endophthalmitis or outcomes of endophthalmitis has not been reported. So that becomes another study. And of course, there could be racial differences could be existing in that particular disease. Say glaucoma is known to have racial differences where African Americans respond differently, Caucasian eyes respond differently, Chinese and Indian eyes all respond differently. So you could say there are racial differences and therefore there is no study done in Indian eyes, say on MIGS or whatever, and therefore we are doing it. So then that makes sense, but you can't just say this is the first study in India. So what is not okay is this is the first study in India. So what? Why do you think it will be different in Indian eyes? Somebody says, oh, this is the first study of 300 patients using Avastin in India. But then Avastin has been studied so many times in the past. So what's different in Indian eyes, right? So that's not okay. This is the first intervention in India. There are a lot of papers which get submitted like this saying, this is a cutting edge laser and I have done this in Indian eyes. So 1 or 2 is not going to cut it. So you say at least 50 eyes. So you say I did this capsule laser or this femto laser in 50 eyes with white cataracts. That makes sense. If you just say 50 eyes, it's not going to cut it, right? A lot of grandiose claims are made by the authors. The hypothesis is sometimes not very scientific, right? So these are not lacuna in literature. Let me take you, I've just already opened this up. Let's see how this introduction has been written in a paper. It's a well-written introduction. So this is from AIOS. It says, talks about characteristics of tear meniscus using a spectral domain OCT in medically controlled glaucoma, right? So we're talking about the tear meniscus in glaucoma patients, which is evaluated using the OCT, right? So let's see what the authors have written. They talk about pharmacological management with topical IOP lowering medications. So they are introducing the term of IOP lowering medications, and there are beta blockers and there are a lot of the other things. Several studies have been conducted to determine the frequency of dry eye symptoms in glaucoma patients and healthy controls. And OSDI, the ocular surface disease index, was employed in majority of these studies. And others have also done using TBUT, Shermer's and all of that. With advancements in technology, ASOCT, including ocular surface epithelia and the tear film, can be now morphologically examined, right? Previous studies have done using OSDI, right? However, there have been limited studies that objectively assess the TM levels, right? Tear meniscus levels in medically controlled glaucoma. So this becomes the lacuna, right? This is the gap. So previously has a lot of people will say, "Arey, too many things have been done," but no, they are actually telling you that there has been limited studies that objectively assess the tear meniscus using OCT in medically controlled glaucoma, and therefore this study aims to objectively evaluate using ASOCT and other things, right? So this is a well-written, so what is known, what is not known, why is it important, and therefore we are doing it, right? So that's a well-written introduction, and that's how you should be trying to write your introductions. There are more examples. Let's go to methodology. It really again depends on the PICOT approach which Dr. Apurva talked about. Let me quickly go to that. So what do you write in the methodology? It's just about PICOT. Who are the participants? Whom did you include? Specifically, whom did you exclude? Is something that you should write about. Then what is the intervention that was done, or basically what was the exposure that was done? So whether you did a drug study which is randomized, non-randomized, etc., or it could be simple questionnaire that you gave, right? A questionnaire was administered and some data was recovered, right? So how did you actually gather data is what intervention means. C stands for comparison group, right? So if there is something that you've done and you have not compared it with something that we are already doing, so we see so many, so there is a session called Under the Apple Tree, and a lot of these kind of sessions do happen, right? A lot of innovation happen and hackathons happen now in ophthalmology especially, but unfortunately none of them is comparing it to what we are even doing existingly, right? So, use this fundus device to image your patients. But then is it equivalent to what I'm currently using? It could be another smartphone based device, but the comparison is not happening. So unless there is comparison, it could be an active or a passive comparison, but that comparison group is something you have to remember and think about even before you start, right? And then you put it down on paper. O stands for outcome measures. So you define it well. We talked about statistics and continuous variables, categoricals, etc. So you define your outcomes in terms of whether it's a continuous, whether it's a categorical, how it was gathered, how it was measured, and how you recorded it. I'll give you an example. Say intraocular pressure as an outcome. You talk about mean reduction in IOP after a particular surgery. That's in millimeters of mercury, right? So that's a continuous variable. You could also define it and say IOP of less than 18 is success and IOP more than 18 is failure. Now that is a category, right? There are two categories, either it's success or it's failure. It's a completely different way of looking at the data. for somebody who doesn't deal with analytics or statistics, it looks the same thing. Array, we measured IOP, we just took and we saw whether this surgery works or doesn't work. But from a statistical angle, it's entirely two different things. One is a continuous, the other is a categorical variable. And I showed you some, when I demonstrated the AI, that's how, it really matters. The type of tests we use, the type of sample size we do, entirely matters. So defining that outcome measure becomes very important. So, you should have a sentence on outcome measure even in your methods in the abstract, and much larger group, even, you could use a subheading and say outcome measures, primary, secondary in your method section. And the type of the study is, of course, you need to mention that in your manuscript, whether you did a retrospective or a prospective and all of that, right? This is how you write the method section. It's useful to use a flow plan to show your methods, even if it's not a randomized trial. So you could use something like this, where I commonly use this. So on the left, if you see, that's where it really is happening. So enrollment, intervention, and analysis. Right? So it starts with informed consent, inclusion, exclusion. Whether it's randomized or not doesn't matter. Maybe you have two groups. If you don't have a control group, it's very difficult to do analysis. It's just a simple description, and the potential of the study is relatively poor. But if you do have control group, maybe 2 or 3, you could use this, right? So this is what was done. Surgery was done day 1, day 7, what was done, 3 months, 6 months, what was done, and analyzed. These are the outcome measures. Right? In one quick go, people will realize, what you actually did. This is something similar. If you look at this, it's relatively complex. It gives you a, gives you nightmare. Array, what do I do now? But then actually, when you read the description of this, it's almost like a 5-page document from the DRCR network study, right? So flow plans really help editors. It helps the readership, and it helps, it improves the publication potential of your paper. Writing the result section, again, it should be initially based on your primary outcome. So mentioning sample size is the first thing you do. So you say we included, so whatever, 70 eyes or 70 patients for the study. That should be in the results and not in the methods. Then you talk about the demographics. The mean age was this, gender, we had so many women, etc. And then you talk about primary outcome, right? If you're comparing between groups, even talk about whether the groups are comparable right at baseline. And always talk about primary outcome as much as possible. I'll give you an example. So when there is a study on phacotrabeculectomy, there are 2 main outcomes, isn't it? One is intraocular pressure, and the other is vision. Which do you think is the primary outcome in this kind of study? Phacotrabeculectomy. Which is the primary outcome? Intraocular pressure or vision? Intraocular pressure. Simple, right? So when I'm reading that result section as a reviewer, the author writes, so there were 70 patients in each group. They were comparable at baseline, and then they talk about vision. Baseline vision was so much, then it ye hua, wo hua, 3 months, 6 months, 1 year. And then there is last one sentence which talks about baseline IOP was so-and-so, and it dropped to so-and-so at 6 months. That's it. There's like one liner about the primary outcome, and that too at the end of the result section. Do you think it is okay? Of course not, right? So concentrate on that primary outcome. That primary outcome really is the gap in literature, isn't it? So that's the gap in literature. That's how you are going to calculate sample size. That's how you are going to write a large section of your methods. That's what you're going to talk about first in your results. And then if you have space, you can talk about secondary outcomes and complications and other things. A quick word on tables and graphs is, especially figures, is they have to be high resolution even when you capture it. So this is a caterpillar hair in the cornea, right? If it's, general camera settings will be 72 dpi, even if it's a, a high quality camera. But then unless you change it to 300 dpi, even when you're acquiring that image, then blowing it up becomes very difficult. It all becomes pixelated. Right? So this is again a caterpillar hair under 40x magnification, and it was captured using that high resolution camera, right? So these images become like at least 8 MB, 9 MB, whereas regular images will be not even 1 MB, right? So you need to get images in high resolution. This is again, high resolution images. Make meaningful graphs. We have talked and we have seen a lot of graphs in using when we are looking at AI and statistics, and I showed you the protocol T and all of those graphs, right? So imagine this graph. This is intraocular pressure in 2 different surgeries. These are standard deviation bars. Without this, you don't know how much overlap is happening and how to actually interpret these. This is this is birth weight and gestational age. Without these lines, everything looks gibberish. When you have these lines, you will see that you are comparing no ROP versus any ROP, and it makes some sense. So when you see the blue line, it is gestational age increases, birth weight is increasing. In the ROP group, after 32 years, gestational age is increasing, but the birth weight is not at all increasing. Now we know that low birth weight is a risk factor for ROP, irrespective of their gestational age. So there are patients here who are born after 34 weeks but have had ROP, right? Because they were very low birth weight, less than 1500. So these kind of graphs will give we've seen how to create these graphs using the AI, but this, even when you're writing and putting it in your manuscript, you should make meaningful graphs. Again, we have seen box and whisker plots before. I'm not going to go into details. Last section we'll talk about is discussion. Remember, it's a very critical part because whatever findings you have, you are trying to fit it into the existing jigsaw of current literature, right? So it has to draw on what is there, and then you are making sense of what is known, and therefore where my new finding adds on. So you initially recap the methods and in short and summarize the positive findings of the study, and that is where the real juice or the amrus is. That's I like to call it that. So if you're actually reading a full text paper and you don't have a lot of time to go through a lot of details, read the first paragraph of the discussion. That will give you the whole, if it's a well-written paper, it will give you the whole gist. So you say this was a retrospective study where so-and-so patients were done, and this is what we found. And then whatever you found, you have to expand on it in subsequent paragraphs, right? And you need to compare outcomes with similar studies from literature, right? And in case of differences, you need to postulate reasons why my study found slightly different results from what rest of the literature is saying. And if you are in agreement, you can mention that, oh, our results are very similar to the previous studies, and of course mention advantages and drawbacks, and then draw conclusions, and always end with future research scope in the field. So the success really depends on how well you have done it, how well you have written your methods, how well you have discussed your results, not really on the results on themselves, right? So this is pretty much what the editors and reviewers are looking for when they pass or fail a particular study. So we don't have much time to go into abstracts. It's an art writing abstracts on its own. It should be informative and not just some description, right? You could say, we did this and we found this, and therefore this is a take-home message. But you could write more information in terms of, we did this. This was the difference in IOP with a p-value, and therefore this is the take-home message. So that gives a far more. So we don't. Lastly, I'll end with checklists. It's really good to write your papers using checklists. So this is the equator network. Let me just demonstrate to you. This is the equator network. Equator stands for enhancing the quality and transparency of health research, right? So what this really tells you is different types of study designs can be written using different kinds of checklists. So these are some of these study designs. Randomized trials is a consort checklist. Observational studies is strobe checklist. Systematic reviews is Prisma checklist. Case reports, Dr. Akshay was talking about case reports. It's written using the CARE checklist, right? So lots of there are more than 600 checklists. So how do you know which, how do you actually choose a checklist to write your own paper? And you can explore this. So these toolkits, you can click toolkits, and then if the internet works, hopefully I'll be able to show you how to get to your checklist of concern. But generally we use these, and all right, so this is opening up. So toolkits, you go use it. Writing research also has some toolkits. Selecting the appropriate reporting guidelines is what we are actually looking for. When you click this, right, select the appropriate reporting guidelines for your article. Complex figure will open up here, which we are not interested in. Then there are many tools you can go and explore this. Till it's opening, let me show you this strobe checklist. There are many, there are 3 types. One is for cohort studies, one is for case control studies, and one is for cross-sectional studies, and one is a combined, right? So the strobe checklist looks like this. Title and abstract is item number 1. Introduction is items 2 and 3. So objective or introduction part 3 is state-specific objectives, including any pre-specified hypothesis. Study design, setting, participants, variables. So you can actually go through these, and if you use this more and more, you will realize that these are some of the non-negotiables which should be part of your paper. So I'm going to show you quickly about how to employ this. So what I do generally is I just copy paste all of these into the Word document before writing anything, right? So this is record 1.1, record 1.2. Record is the same for strobe for retrospective studies. So this is an abstract. So this is all covered. So this is a study on utilization of sutured versus sutureless scleral fixated lenses that we use for aphakia, right? So records or Strobe statement 2 talks about explain the scientific background and rationale for investigation being reported, right? So I'm just going to uncover this. So initially you talk about damage in the zonules and other things, and there are many different types of intraocular lenses and multiple options exist. Then we talk about SFIOL. Remember that we are zooming in from the larger context to the specific context. So we talk about aphakia and different options. Then we talk about SFIOLs, and there are two main types. One is scleral fixated, sutured and sutureless. The problem statement is then that sutured SFIOLs are a little difficult to do and all those other things, and potential solution is this sutureless SFIOL, right? So it's technically less demanding, it's quicker to perform, has less complications. You can talk about your settings. So the whole idea is not to go through each and every item, but to show you that you can actually copy paste each of these. Let me show you. So you copy paste each of these, and then you can actually present key elements of a study design early in the paper, describe settings, locations. So what you can actually do is just copy paste this, show it to ChatGPT, and say this is a study that I'm writing about. How do I answer this question? And it will actually give you 3 or 4 sentences. Each statement will have even 2 sentences that the entire paper is ready, right? So you can actually answer 1 question a day based on what you have done. So in 20 days, your entire manuscript should be ready. If you are using writing softwares or others, generative AI like ChatGPT, that could be even faster, right? So you can actually finish writing papers in like 2 weeks or so, right? So just asking ChatGPT versus asking ChatGPT specific questions directed towards your study design, or your title. You say, oh, my title of my study is this. How do I answer this question about setting, location, relevant dates, etc.? And you of course provide those, right? This is my setting, the Toshikere Center in South India or whatever. And then you say, location is Western India, Northern India, relevant dates, so Jan to December 2024 or whatever. And what was the exposure, how long follow-up we've taken, how we did data collection. And then ChatGPT will actually give you the entire thing in a very polished fashion, which will not be looked at as plagiarized. It will not be caught in plagiarism softwares and AI catching softwares. And so each question you can actually post to ChatGPT and tell it that this is my context. These are the possible answers to these questions. Now help me refine it. And then you will get answers to these questions. And it should not take you much time to tell what is your setting, what is the location, what are the dates of your study. And then you get like 2 or 3 sentences based on your inputs, right? So that is like prompt engineering. People call it prompt engineering now. So you are improving your prompts. So you can use these checklists to write prompts on AI software and actually write an even more polished paper, right? So that's pretty much what I wanted to share about how to write good papers. You can use some software like Grammarly, that actually integrates with your Word document and helps you write slightly better. You can use software like Zotero for writing your references. And lastly, you can actually use some of these journal matchers. So you can just let me show you one of these. So Web of Science journal matcher, Elsevier journal matcher. So the Elsevier journal matcher may be slightly better. So this is where do I submit my paper, right? So that's what it is. So you need to put your title and your abstract. Based on that, these softwares, which are also AI driven, will suggest to you, where you should perhaps submit your paper. Which journal is more likely to take your paper. So instead of submitting and then getting rejection after rejection, you can actually choose these journal finders, right? You can just Google them up. The internet is slow, so it's not showing up too well, right? So use these journal matchers. It's there on my LinkedIn profile and all that, so you can go through those. These will help you actually zoom into the journals and then submit your papers to the right journals, and therefore expect more acceptance than rejections, right? So that is pretty much what I wanted to talk to you about. If you have any questions, any comments? You can. So basically in Julius, you can you can right click and you can save it as a let me show you some that I have saved. You can actually save it as PNG file or a JPEG file, right? So some of those that I showed you, I don't remember. No. They are just JPEG files, so it's not going to really show. Let me try and see if I have. So this is the this is the one I showed you, right? This is made by. So this is from the protocol T, and this is on right clicking. It will say save as, and you can say JPEG or PNG, and then you can save it to that folder, and then it will get saved like this. And this is high resolution R image, which can straight away be submitted to the along with the manuscript, right? So that's relatively simple. Yeah. Yeah, that's what so it depends on how large your sample is and what is the study in under question. So I'll give you an example. So people have done a study on C3R after for keratoconus and microbial keratitis. And so there is a study published in Konya journal from Spain or somewhere where they talked about 30 cases in like 6 years or something. It was published in Konya. And somebody in the crowd said, "Oh, 30 cases. I have seen it in 1 year. And I'm from this organization and I've seen 30 cases like in a year because they all get referred to me." So then, that's a huge difference. So I told them, so how many in 5 years? So they looked up their data, like 280 patients. Post C3R, bacterial keratitis is rare, right? So even 30 was large. But now this person is coming and saying, "Oh, I have 280 patients data." So then that becomes really large. Right? So for a rare disease, even little bit of, it is good. But for a common disease, then you have to be substantially large, and then your statistics have to look at slightly different ways. So somebody, say, intracameral moxifloxacin reducing incidence of endophthalmitis has not been studied in a prospective randomized trial, which is fine. The largest studies, so you need to actually present odds ratios and say the reduction, the risk of endophthalmitis is like 2 times lower or 5 times lower or whatever that is. That has not been done by any of these studies from Arvind, which has looked at 5 lakhs, 10 lakhs. So, there are papers like 20 lakh patients, but then they don't offer odds ratios. So you could actually say, oh, we have data from 40,000 patients where, we used some and we didn't use some based on institutional patterns. And now we find that actually rate is lower and it is lower by like 3 times or 30% lower chance of what. That makes a lot more sense. So your numbers may be even smaller, but your statistics are different. So then, it is a mix and match, not just large samples, but, anything which is novel statistically could also make sense. So if it's in a nutshell, if it's a rare disease, even doubling the number could help. And if it's a common disease, I would look at not just larger numbers also, but looking at it from a statistical angle and creating novelty there as well. I think we are short of time, so we have finished this session, and hopefully, it has made some sense and given you a framework to go and do your own research. So thank you so much, and to everyone who attended, thanks so much.